Okay, so to start, welcome to our wetland section plenary. So I'm Aleck Wang from WHOI, associate scientist at Marine Chemistry or Geochemistry Department. And so I'm studying carbonate chemistry, ocean acidification, blue carbon, develop sensor technology for measuring carbon parameters. So I'll first welcome everyone. This is the first in-person meeting, after COVID. Pretty exciting. And I'd like to acknowledge my co-chairs, Jaime Palter from URI, Xinping Hu from Texas A&M, Corpus Christi, Maria Tzortziou from City University New York, and Jeff Bowman from Scripps. So a little bit of history. So back a couple of years ago, 2018, we had the first lateral carbon workshop organized by Kevin Kroeger, USGS here, and some of the other organizers also in the room. So at that time, the workshop tried to identify the major key gaps, knowledge gaps on the lateral carbon exports. So since then, okay, good. Since then, there's some pretty good development, and especially for the last few years. So we think it's maybe a good time to update people in this field, particularly in the context of blue carbon. So this is kind of the motivation slide. So we all know the wetland is very productive, and per unit area absorb a lot of CO2. So for those CO2 absorbed, we can on the upper left we call it the CO2 pump, wetland CO2 pump. So pumping a lot of carbon dioxide by the plant's photosynthesis, and some of it return back to the atmosphere through CO2 exchange in the tidal water, also in the sediment, and then part of the carbon being stored in the soil. And those are the kind of the traditional blue carbon. Okay, so there's quite a bit of carbon being exported through tidal exchange to the coastal water. So in recent years, people start to argue that whether we should treat those exported carbon as blue carbon as well. So the recent synthesis suggests this export is pretty big. It's some suggest more than 50% of the annual production, net production, being exported as various forms of carbon. So the reason we argue that maybe it's treated as blue carbon, for example, DIC, so it's in the form of bicarbonate, and there's some alkalinity as well. So the residence time of DIC or bicarbonate is pretty long in the ocean, right? So it's thousands of years. So so we think if those carbon being exported to the shelf and further maybe into the deep ocean, then they should be treated as the wetland storage, right? So for DOC and POC, depends on their how quickly they recycled. They may also store somewhere in the ocean. So that's where this concept of mobile blue carbon coming from. So we're gonna have. So this is a little structure of the session. We're going to have 5 plenary speakers, and then cover different aspects of it. So I'll give you some latest development. So 4 talks are going to be before lunch, and then 1 after lunch. And then after that, we're going to have 5 lightning talks. Okay. So then followed by panel discussion. So I think we're going to just follow the previous format that, if each plenary speaker has some time left, we're going to ask questions. Otherwise, we're going to ask questions at the end. So I'm going to have few slides to maybe still stimulate the panel discussion at the end. So that's it. Again, hopefully, hope you will enjoy this session. So I will have the first speaker, Santos from University of Gothenburg. And he's going to talk about the global overview of.
Yeah, all right. So today we are going to kind of change the perspective rather than thinking about kind of top-down fluxes in the ocean. We're going to look at lateral fluxes from the continent to the ocean, asking this question: how much of that can actually get all the way down so that we can call it carbon sequestration? So I'll be kind of providing an overview of the evolution of these blue carbon fields with a focus on lateral fluxes, the dissolved components of carbon coming out of mangroves and salt marshes. So just before I get going, my talk today is going to be mostly about the work of those four people. On top, Gloria Reifmeyer, she's a postdoc, Yvonne and Alex, PhD students working on the topic, and also Xiaogong Chen in China, another postdoc. And I'm also going to talk about this paper. That's a perspective paper that came out last year with a bunch of our colleagues working on the topic and many collaborators over the years. So there is lots of conversations about what's blue carbon. So I realize many of us here are thinking about, the carbon pump in the open ocean. And I think this blue carbon term has been kind of hijacked by more of biological coastal community, thinking about mangroves, salt marshes, and seagrass beds. So that's roughly what we want, to be able to call this ecosystem blue carbon ecosystems. They gotta have an ability to uptake greenhouse gases from the atmosphere. That's the first criteria. The second one has to be long term. You cannot pick up CO2 from the atmosphere and let it go really quick. So you're talking about time scales of several decades or longer. And number three over there, you got to be able to do something about that. So the whole community likes to visualize that. You got to be able to do things like maybe changing the tidal flows, or maybe removing these barriers, or maybe growing trees, whatever you want to do. So those are the three key criteria for calling an ecosystem blue carbon. And you can see that pretty much the whole ocean, would fit that definition. The whole argument about the blue carbon has started by comparing those three ecosystems: mangroves, salt marshes, and seagrass beds, to terrestrial ecosystems. So if you look on top over there, we have a very small area of those intertidal or subtidal systems like seagrasses, but the carbon sequestration on a square meter basis is much higher. So you have much more on a square meter basis for salt marshes, mangroves, and seagrass beds. If you multiply the top two graphs, you come up with those numbers. So people have been saying for years now, for maybe 10, 12 years, that those three ecosystems, they are, can be better than tropical ecosystems. There is lots of issues with that argument. One of them is that those three ecosystems are depositional environments on the coast. They're actually capturing some of the carbon coming from the forests upstream, right? And there is more issues, but anyhow, that has been how the community has been developing over the years. So if that vision, that soils or sediments, whatever you want to call it, depending on where you're coming from, in those three ecosystems, they're super efficient at sequestering carbon. So people have been sampling soils and sediments of those ecosystems for many years now. So they occupy a very small area of the global oceans, but they disproportionately contribute to the accumulation of carbon in sediments. That has been another key argument that has been flying around, and you can also, agree about that. So with that, the whole community has been going through a bit of a race, to take sediment cores. So, right, my own group, we have taken hundreds of those sediment cores all over the place and trying to characterize how much carbon is there from a storage perspective and also from a flux perspective. So we now have these global maps of soil or sediment carbon. This example is for mangroves, an exercise that came out a few years ago, and we know how much carbon is in storage. We also know, as of last year, how much carbon is fluxing into, is penetrating those sediments in mangroves and salt marshes. So that's a global map based on a bunch of sediment cores all over the planet, including the hotspots, which would be the Pacific Coral Triangle, somewhere there. That's where there is lots of carbon accumulating, especially in mangroves. So we now have those global numbers. We have inventories, fluxes, and stocks. We even have predictions. So we kind of have predictions of what's going to happen with salt marsh and mangrove carbon fluxes with climate change. So because those ecosystems, they kind of like the heat, they like rainfall, and they're okay with sea level rise. They tend to keep up with sea level rise as long as there is a space behind them. So with climate change, we should have more carbon sequestration in mangrove and salt marsh sediments. So we have had this strong focus on looking at those sediments. So that's a conceptual model that came out in Nature last year. There's been this big fuzz about the blue carbon concept, and there is this big emphasis or this perception that blue carbon is all about sediments, right? So I think that our community has been, we have been digging our heads in the sands. And I think you've got to go beyond that. And the main message I want to bring today is that by going beyond, you've got to look out to sea. All right, so that's my main message today. Okay, so this whole thing of looking out to sea has began, many years ago with this review paper by Steve Bullion that he pretty much put together this pie. So the pie, you can visualize that as the size of the global primary productivity in mangroves. And, the slices of the pie represent the fate, what's happening with all this carbon that the trees suck up from the atmosphere. So we have DOC exports, POC, emissions back to the atmosphere, and burial. So from this blue carbon perspective so far, we're only looking into burial as the carbon sequestration mechanism. So what about the half of the pie that has been unaccounted for? So that's what we're looking for. And our hypothesis has been that alkalinity, bicarbonate, and DIC is pretty much this pie, and have been working on this hypothesis everywhere. The other key paper that came out in 2016 by Aleck Wang is showing a full budget for US East Coast saltmarshes, where there is a clear comparison between carbon burial and really breaking down the multiple components of outwelling, lateral fluxes. So in this case, for US saltmarshes, DIC is much greater than organic carbon burial. Now, the question is how much of this DIC is actually CO2 and how much is as bicarbonate. So that's the next step in our journey here. So this is a crazy schematic that summarizes, how carbon budgets should look like in this blue carbon ecosystem. So we start with CO2 uptake by vegetation, right? They're pretty good at that. Some of that stays in sediments, and that's what I'm saying that we have been doing that for a while. We kind of got a good handle on how much carbon is staying locally, right? So the outwelling concept is actually quite complex because we don't know how far this carbon is actually going. So we do have the DIC, the DOC, and the POC going away from those ecosystems. Part of that is actually going to return to the atmosphere along the journey, and only a part of that is going to stay in the ocean as bicarbonates, refractory DOC, or maybe even accumulates, in sediments offshore. So that's the carbon that we want to see. That's the carbon outwelling that becomes carbon sequestration. Right? So in the same paper, we kind of put together a back of the envelope budgets, trying to put in perspective those numbers. The challenge is that many of those studies, they are kind of isolated. They quantify only one component of the carbon budgets. Very few studies, they integrate all those fluxes. So you're kind of taking average for those different components from different locations. And there is only a few sometimes. For example, seagrass beds, we don't really have any estimate, even at the local scale, of DIC outwelling. Prove me wrong. Maybe somebody's cooking up one of those data sets, but that's not out there yet. So from that overview, we kind of found out that in pretty much all of those cases, outwelling tends to be bigger than carbon burial. So by looking down to the sediments, we're missing a big picture, a big part of this whole picture. The thing about outwelling, it's all about this fraction of how much of that carbon can stay in the ocean, how much goes back to the atmosphere. So at the end of the day, that's going to be a function of the mechanisms producing just carbon in the blue carbon ecosystem. So we have this diagenetic reaction going from a simple one-dimensional system where diffusion dominates transport. And we also have this vision in mangroves and salt marshes for sure, where we have all these crab burrows that they make those sediments, they feel like a sponge full of holes. So as you know, sponges, they are pretty good at taking water and let it go. So when you have this three-dimensional structure with those burrows, we have lots of space, a bigger area that enables those diagenetic reactions to go, beyond simple oxygen respiration. All right. So in mangroves and salt marshes, we have sulfate reduction really dominating, carbon oxidation in the soils. The thing about sulfate reduction is that it actually produces alkalinity or bicarbonates, rather than CO2. So if you're doing that, we're essentially converting organic carbon into bicarbonates, and that gives us sequestration. If you have only oxic respiration, we are just returning the CO2 back to the atmosphere, and that's zero. No gain from a carbon sequestration perspective. So those places are full of those burrows. That's a photo from one of our earlier papers. I had a few students that didn't like it very much, and I asked for them to do that. That was really fun, but trust me, that's one of the hardest jobs we can do. It's a cost. You put some resin in the sediments, and you dig up, all right? So that's a lovely experiment, after a couple hours you hate it, but you can see that three-dimensional shape that enables the flow of water in and out of the sediments. And now we are pushing everything the microbes like, oxygen, sulfates. There is plenty of carbon in those sediments. So we are kind of driving those chemical reactions in the sediments. All right, so we have also quantified the fluxes, how quick pore water is exchanging in those sediments. The way you do that is using radon, geochemical tracer, because that integrates the ecosystem scale fluxes rather than going for an individual crab burrow. So we did that in Australia. We had 6 sites going from the tropics to Melbourne, temperate mangrove, shrinking almost with salt marshes in the back. And we modeled pore water exchange rates in those places. So to give you a scale about that process, here's the data from the 6 systems. We have rates going anywhere between a couple centimeters per tidal cycle up to 20. So a centimeter per tidal cycle, that means that we have, just much of the sediment pore water exchanging with the surface. It's a filtration process, right? So if you believe that those 6 locations in Australia represent, global mangroves, we come up with those numbers. Those mangroves would be filtering the whole volume of the continental shelf every, 100 or 200 years. So the volume of exchange is equivalent to 1 to 12% of the tidal prism. So if you imagine you have a 1 meter tidal range, 1 to 12% of that tidal range penetrates the sediments. There's something in there and comes out. So you are treating sediments as a black box and seeing what's coming out. So the methods to measure this stuff they can be quite challenging. The method that gives us the ecosystem scale fluxes that I like these days is radium isotopes. So I'll give you a very quick overview about radium isotopes and talk about some of the challenges. So the approach here requires us measuring radium away from the source. So the source is 0, the coastline. And you take a vessel and you go offshore. In this case, we went 12 kilometers offshore. We are looking for a gradient in concentrations away from the source because you rely on the radioactive decay of radium to estimate the physical transports away from the shoreline offshore. And at the same time, you've got to measure the carbon species. In this case, DOC, alkalinity, and DIC, and you got to have a gradient. So, coastal systems are messy, so those gradients are often messy as well. In this case, we got some type of gradient, and we can apply some models and equations that gives us the offshore flux across the shelf. So that gives us an integrated ecosystem flux, everything that's coming out from the coastline. The second methods are time series, usually at the mouth of those tidal creeks, those ecosystems, especially mangroves and salt marshes that are quite suitable for that. So in this case, we had 8 days of data measuring things. Some of them we can do in an automated fashion, like radon, CO2, methane, salinity. On others, we have to fill up bottles, like PLC, DOC, DIC, and alkalinity. So what we do in those systems is to go through full tidal cycles, and we can monitor the water going in, and the water comes out. And we measure the concentrations every minute or every hour, depending on the parameter, and then we can get the fluxes in and out of those systems. So that's the type of data we got, because to get one flux estimate, we need to do either a whole transect. So this whole transect offshore, which is a few days of work, on a vessel, gives us only one outwelling number. And if you do just multiple tidal cycles in a creek, we also only get one outwelling flux estimate. So it's a lot of work to get one number. Right? So we are building budgets, under exchanging that, usually we only have a few days of data. Right? And because those are isotopes, those few days of data, they tend to integrate over multiple days as a function of the half-life of the isotope you're using. So the carbon budgets where you can actually quantify all of those fluxes in the same system, they kind of look like that. This is a budget that just came out in LNO by Yvonne and Chagong, where they estimate carbon burial, and we can compare that to outwelling. So this is our conventional blue carbon, 96, versus alkalinity outwelling, 78. So we're talking about the same order of magnitude in this case. If you only look into that, we're underestimating the carbon sequestration ability of those ecosystems, because this alkalinity should hang out in the ocean for thousands of years. All right, another system in Australia where you quantify those processes. In this case, there was plenty of alkalinity, 4 times bigger than burial in this mixed mangrove salt marsh system. That's a paper that just came out at the end of last year by Alex Cabral, where he actually tried to combine those methods and look across different spatial scales, going from the groundwater scale, which would be like, the mangrove, the burrow, the crab burrow, into the tidal creek scale with a time series, going offshore, the estuary, and then going offshore out towards the shelf break. So the fluxes at each of those compartments are different, but in this case, we could quantify them at about the same time, the 2 or 3 weeks we had in the fields. So in this case, in the Amazon, those mangroves at the more local scale are producing about a third in terms of alkalinity versus burial. As you go offshore at the continental shelf scale, the burial signal becomes more important. We're talking about 19 versus 2. And that's because you have algae burial that's coming from multiple sources beyond the mangrove that we cannot actually quantify. All right, so right now, Gloria has been working on this compilation of algae DIC and alkalinity data in salt marshes. Some of you are part. Here is a list of all of our colleagues who are providing data. They're trying to build a global view of how important this process can be. So we now have DIC and alkalinity data from those sites. It's about 40, 50 sites, depending on how you count them. Lots of mangroves in Asia, a bit in Africa, South America, Australia, and just a few salt marshes that actually have alkalinity data. There's plenty of DOC and PLC data, but very little alkalinity. Right? So one of the key questions that Gloria is asking is whether this outwelling is going to have implications to the pH. So to test that, we are using the TA to DIC ratio as a proxy. So if that's above 1, that means we have a bit of excess TA, and then therefore the interpretation would be that this outwelling is going to be a bit of a buffer against local ocean acidification. I would call coastal because it's a local process. If the ratio is below 1, we have excess DIC, therefore we are acidified the environment nearby. So on average, those ratios in mangrove surface waters and in salt marsh surface waters, that's pore water and that's surface water, they are usually higher than 1. So it's often site specific. Sometimes they can be buffering, sometimes they can be acidifying, the immediate coast, but on average, they tend to acidify. There is more CO2 in DIC, and the alkalinity is not really compensated for that. And that's interesting because we had data before from a single system in Australia when we started thinking about this hypothesis. And that was a mangrove system, that's the mangrove area, where we did a survey offshore of pH. And we do find that inside the mangrove, the pH is low, right? So, those inputs from pore water are acidifying the local mangrove waters. However, as those waters mix offshore, the CO2 escapes to the atmosphere, but the alkalinity is hanging there. So that alkalinity is actually creating a signal offshore where the pH is slightly higher than if you go further offshore where you don't have the effect of mangrove waters anymore. Right? So you do have those site-specific impacts. I think on average, there is more of an acidifying role, but sometimes can be buffering against local acidification. All right, so the other thing that Gloria has been trying to do is to try to look into some factors that you can use to model global scale alkalinity and DIC outwelling and go beyond burial. So the factors we are trying to use for now are temperature, precipitation, sediment accumulation ratios, tidal range, carbon accumulation, and location. All right. So the data sets we have, you can see the dots, you can count them. There is only, 15 or 20 estimates of outwelling. And the challenge here is that the estimates of outwelling usually are on the scales of a few days or a few weeks. There is only a few, maybe 2 or 3 seasonal studies. And now we are trying to explain those, kind of short time scale observation as a function of longer time scale processes like carbon accumulation rates that takes place on decades or hundreds of years, right? So we haven't really been able to find a clean or a single factor that explains outwelling out of those obvious proxies that we have here. So because there is not a single variable that you can use to model or to upscale, our best guess models right now are simply an average of all these studies that tend to focus on individual components of the carbon cycle rather than integrating them. So if you try to update the earlier budget, the pie chart that Steve Bullion put together 15 years ago, that's what we come up with. So the global burial rates in mangroves and salt marshes seems to be much lower than the exports of both DOC and DIC. So outwelling, is a bigger player from a carbon budget perspective. We don't know how much of this DOC is going to stay in the ocean. We don't know how labile this can be. So we cannot even make a guess for that. But what we have is that the DIC that breaks down into CO2 and alkalinity, the alkalinity component seems to be higher both in mangroves and in salt marshes. So by only using carbon burial as the argument for saving those ecosystems or preserving them or, for carbon credits, we are really missing a big part of this picture. So just to wrap up, this blue carbon science has been evolving quite quickly. The term was coined in 2009, so it's only like 12, 13 years. The sediment business, I think we have kind of reached maturity with the sediment business. We have global estimates. We know what's driving that. So we know where it's going. Right? So the outwelling process we're talking about, it's complex, but the alkalinity component of that outwelling, for sure, it's blue carbon. Right? It's carbon sequestration on long time scales. Groundwater exchange or pore water, you name it, depending on where you're coming from. But that's something that's coming out of the sediments via advection. So this groundwater exchange processes is linking those two carbon reservoirs. And we think that this outwelling is bigger than burial. So considering alkalinity really changes the game if you're talking about carbon budgets in those systems. So I will stop here. Thanks for having me, Aleck, and other organizers. It was a pleasure to be here today. You have questions now, or should I sit down? Huh?
We have one question online here. And there's no name, so it's an anonymous question. Do you think seagrasses would have a similar effect to coastal acidification as mangroves and salt marshes?
Oh, here you go. Listen, I think seagrasses, they're uptaking CO2, so that does have a local impact on pH for sure. Now, the question is whether that seagrass is actually exporting alkalinity, and we really don't have any numbers on that. So I think that's an ongoing discussion, and we're not settled on that.
I have another one too. Actually, two. They're coming in on the Zoom. What's the difference between groundwater and pore water outwelling? This comes from Songji He.
Yeah, that question comes all the time. The thing is that when you think about those two terms, different communities tend to visualize those two terms in different ways. But from my perspective, when I'm using tracers, like my geochemical tracers, radon and radium isotopes, they're actually tracing any water that's in contact with the sediments. And those sediments, they're giving life to salt marshes and mangroves. So I actually don't care. It doesn't matter. It's just a semantics problem, because the reality is that that water is passing through those organic rich sediments and picking up the carbon that initially was taken from the atmosphere by mangroves or salt marshes. So that's what we're tracing. It was the carbon from the mangroves and salt marshes. So in this context, it's actually, it's a bit irrelevant, even though we can have a really interesting conversation about that.
Any in the audience? I have two more online here, so.
Yeah, I'm Wei Jun Tai. First, thank you for working on this issue that's been interesting to me for years. So there's one comment or question is, we know the ocean alkalinity budget is not balanced. There are more carbonate buried in the ocean than the river supply. And so people have been searching that alkalinity, where it come from. So do you like to speculate with this alkalinity outwelling thing could be is that even close?
Yeah, listen, we know that there is a big missing source of alkalinity, and there was the most recent update was Jack Middleburg's paper last year, right? Yeah. And he clearly shows this missing alkalinity source that's probably on the coast. We know the river fluxes, but I'm pretty sure this is part of the story. If that's going to explain the whole thing, I don't know, but I'm pretty sure that's one of probably the many alkalinity sources that may be hiding on the coast that we cannot really quantify on a global scale yet.
I see the river supplied, alkalinity, but that was geologically inert. So maybe that's not at least not a complete solution. So this could be something. I don't know if the size really goes that high, but it is something. Yeah.
Yeah, I agree. I'm pretty sure that's part of the story. How big this is. I think are working on that right now.
Melissa Melendez from University of Hawaii. I was wondering if you have seen any correlation with the morphology or the physical characteristics of these bays, or compare that with the DIC to TA ratio.
Yeah, there you go. So we haven't tried that. I was talking to Gloria actually this morning. She's the one driving this project, and we actually don't have a simple way of defining. How do you define the morphology? How do you put a numerical value to a mangrove? I actually don't know how to do that. I can speculate or talk about my gut feeling is that would be too hard at this stage because you have only a few measurements. So finding that type of relationship maybe requires, much more data because, the key, variables that we expected to explain outwelling, they are not explaining right now, right? So, but I think morphology is a natural candidate. I don't know how to quantify that to build a correlation. Maybe you have some ideas.
Hi there. This is Lauren Cook from Rutgers University. This is a really great talk. I learned a lot in terms of like salt marsh and mangrove carbon cycling. I was curious, you had a figure that was looking at RCP 2.5 and RCP 8.5, and I think the carbon sequestration potential I think that was what it was. But there was quite a large uncertainty on RCP 8.5, more so than RCP 2.5, and I was wondering what the influences that uncertainty. Is it, the fact that we just don't have enough? We're working on getting more constraints on these systems, or if you could speak to that at all.
Yeah, I'm trying to go back, but Jesus doesn't like me anymore, so I didn't like my talk. But that paper was led by Aleck Wang. It was just compilation. So I remember talking about that, and essentially it's a propagation of uncertainties over time, right? So those models, they do have uncertainties in all those key outcomes in terms of precipitation, temperature. And the mangroves and salt marshes, they also have uncertainties in how they respond to precipitation and temperature. And then you go beyond the mangroves just growing and being happy to sequestering carbon. So you have just multiple steps along the way, and those uncertainties, they get compounded as you go further into the future. So yeah, we may have to sit down and have a look back to the paper to reflect a bit more about which ones are more important. I actually don't remember, but that's a compounding effect going beyond. Heather?
I've got one more quick one while you switch the speakers over. Raiza Shiel from our virtual world. Hi, Professor. Do you think mangroves from microtidal environments would also have a significant outwelling of alkalinity in DIC?
There you go. So this tidal cycle, I really like tides, and I think they're a big driver of those things. So we have found that on a local scale, if you have a single mangrove or salt marsh, both, and the tides are changing over time, and you're quantifying outwelling for each tidal cycle. So inside that mangrove, there is a very, very clear relationship between the outwelling and the tidal range. But now if you kind of take an average tidal range for the whole ecosystem, you go to the global scale, the relationship disappears, right? So we have measured those things in a few microtidal systems, and we found plenty of outwelling, so it's there, right? And even no tides, the outwelling is going to be there. It's going to be much less, I think, if you have no tides. But you do have those systems in some microtidal environments, and they are producing alkalinity, as much as they can. The area becomes smaller because the tidal range is what creates a big mangrove, a big, salt marsh stand. You got to have tides to go big. But on a square meter basis, maybe they're not that different. Thank you.
All right, so continue. That's a great talk. Yeah, I have another question, but we can save it to the panel, I think. but I think actually we should treat a little bit more than just alkalinity, but that's more of a dynamic question. So I'm going to continue this journey, and I'm going to talk about the lateral exports of specific DIC. And I'm also going to show a little bit of new study on the vertical CO2 exchange in tidal water. So I would like to acknowledge my co-authors. Some of here, Sophie is in the audience. This part of this is her thesis dissertation. Megan and Adrian are from USGS. Kevin Kroeger is the lead of USGS wetland carbon program. My lab people, Kate McCaskey and former student Mallory Ringham, and our collaborator, Joe Tomposki, who wrote a paper with us as a postdoc here, and then he's an assistant professor at Old Dominion University. And Shuzhen Song is postdoc at Shanghai now, but he did the recent vertical CO2 fluxes. So on the left is just a quick overview of what I'm going to talk about. So the blue circles are the two fluxes we're going to define in this talk. So the lateral DIC part, and then there's a tidal creek evasion or efflux of CO2 from tidal creek and also inundated marsh area. So I'm going to focus on the 3+1, I guess. Three methods, and we applied the three methods at the same location. We tried to do a cross-method comparison and hopefully this can help us to get more confidence to define the lateral export. So the CO2 effluxes, even though it's not part of the lateral, but I think there's some new findings. I think it's quite relevant to the lateral export in terms of the control of it. So I start with some basic DAC alkalinity chemistry. Aleck already mentioned this, the wetland system export both DAC and alkalinity, and DACs come from the aerobic and aerobic respiration, and alkalinity generated through anaerobic respiration, like sulfate reduction, right? So that changed the tidal water chemistry quite a bit. So this is kind of the example of it on the right. There's a vertical is the buffer factor, beta H, which is the definition on the left. So it's a traditional view of the buffering factor. The higher the buffering factor, the higher the buffering capacity. And the x-axis is the DAC concentration, and this is versus the pH. The contour lines is the alkalinity contour lines. And then notice there's a buffering capacity minimum around 7.5, okay? And that's where the most of the carbonate ion being consumed. So on the left of this contour lines, basically you have ocean acidification going on, kind of situation where you're adding DAC to the system. You basically consume carbonate ion, decrease the buffering capacity. At some point after you reach the minimum of that, the system converted to a bicarbonate buffering system, which where more DAC being added, but the buffering capacity actually increased. So, and then you can see this is the tidal marsh system, and the tidal marsh add DAC to the system, lower the buffering capacity at the beginning, but at some point, passing the minimum, buffering minimum, and then actually the buffering capacity increase. And this is the springtime. And in summertime, you can clearly see that, in addition to DAC addition, you have alkalinity being added to the tidal water. That's where the tidal cycle actually, the alkalinity did actually show that there's a cross contour lines addition of alkalinity, and then the resulting buffering capacity actually higher than where the tidal water comes in at the beginning. All right, so the three methods of the first is the time series. So the time series is a little bit different than what the ISAC did there. This is we set up a time series station at the Tzortziou town, which is not far away from here. This is a micro tidal site. There's limited river input and not much of nitrogen loading, and limited groundwater input. So at the time series station, we basically deploy off-the-shelf sensors. Those are the YSI XO type of sensor, and then we measure water flow with ADCP. We take bottle samples over tidal cycles, and then to verify so basically we have two time series approach. One is try to use bottle samples and the off-shelf sensors to come up with a high-resolution DAC flux. And the other is Sophie did her PhD thesis on the channels, which is in situ DAC sensor, and we use that to verify the other time series method. I'm going to show you in just a minute. And then there's a very important aspect, I should say. The hydrodynamics of the water is very important to define the lateral fluxes, right? So in this case, we use the hydrodynamic model to correct the overland flow, which is not passing through the time series station. And then we also correct the groundwater input at the site. So the first method is called a multilinear regression. So it's quite simple. You have bottle, we basically treat the MRI model with sensor measurement and combine the MRI model with the ADCP and the hydrodynamic model to come up with the instantaneous DAC fluxes. And on the right, this is the basically 2-3 years of in situ DAC model of in situ DAC concentration along with the DAC fluxes. So the negative number is going out to export. Okay. So then this the lower right is kind of seasonal variability of the fluxes, and then the you can clearly see the spring neap tide cycle as well. And yeah, okay. So you can integrate all the instantaneous fluxes and come up with the time scale you want to see. In this case, this is the monthly mean fluxes, and you can see the high input export in the summer, and then in the fall, summertime is about 40% of the total flux, and the fall is 35, and the annual flux number is about 400 gram carbon per meter square per marsh, and between January and March, we basically treat at 0 because system are mostly frozen. Okay, so how do we know that our MR method is correct? So this is to verify that method, we deployed our kind of first generation channels sensor there. So this is basically we deployed a sensor right at the mouth of the creek. Okay, and this on the right is the time series example of one of the time series we get in the summertime, and the concentration is blue, and the orange is the modeled concentration. And so there are some places that match pretty well, but there's other places that may not. But it's not surprisingly, but actually the modeled DAC fluxes match the channel DAC fluxes pretty well. The main reason is actually the arrow, even though there's a mismatch between the model and the sensor, the most of that is random, and then because the flux is negative and positive, so actually the total flux cancel out each other. So this is a kind of summary of the comparison. 2 periods. One is in the summer, one is in the fall, winter, and both. This is a corrected number in the gray. The two methods basically match each other pretty well. So we are pretty happy of this, and the high resolution measurements also can tell you something, much more detail. In this case, how the DAC fluxes may vary over individual tides, right? So this is the histogram of DAC export flux. The vertical axis is the number of tides, okay? And then so the 2 periods, one is summer, winter time, and then the center of it is the percentage number of tides is mostly in the near, the flux of that is actually close to 0. So essentially the tide goes in to the marsh and then comes out. The net flux of that is not too far away from 0, okay? So imagine you go out there, this is kind of the typical tide, right? So if you go out there, the most of the chance that you're probably going to catch this in the middle of this histogram, right? So, and then you miss the parts on the both sides. The true mean actually is on the left side of this distribution. In other words, it's a little bit higher than the typical tide export. And then, so that's one of the reasons why we care about the high resolution measurement, because you could significantly obscure your mean flux calculation. So this is just a little summary of the two methods. The DAC export highly variable over wide range of time scale, and DAC export highest in summer, and then fall followed by fall and spring. This coincides with the marsh production. The hydrology, water fluxes play a very important role, okay? So we need a high frequency measurement because, you know, there's a good chance that we may miss quite a bit of signal if we don't do that. If you don't have the sensor to do it, you could use multi-regression and proxy parameters to do that. We show that it's reasonable. All right, so good. So the third method, or second method, I would say. This is a new paper that's come out last year. And this method is basically based on the radium and thorium core sediment, okay? So I can play a little bit cartoon here. So we know that this is a Joe Chambosky's work, so he cannot make it. So hopefully I can represent you know, his work well. So the thorium we know is particle active, so it's pretty much bound to the particles, right? So but its daughter, radium 224, is relatively soluble, right? So when you have exchange like vertical exchange or lateral exchange, that will cause the disequilibrium between the radium and thorium 228. So this is the kind of the representation. So the surface layer could, because the deficit of that ratio is probably because the vertical exchange, and at the depth that could be due to the horizontal export. In this case, the export of water from sediment. So we can use that method, apply that method, and then and by measuring the DAC profiles as well as the activity of the two isotopes, then define the lateral flux. So this is what we did. In this case, we did actually two sites. One is the same site as I showed the time series before, and then the other side is actually a little bit south of the first site. And the two sites is very similar in terms of tidal range and other vegetation. And then we did kind of transect three sites perpendicular to the tidal creek. And this basically tell us a little bit more what is the spatial variability of the fluxes, and then, you know, even on the same system, whether there's a you know, change of horizontal fluxes. So here's the upper plot shows the water exchange from two sides, blue and red, and then the three stations, creek, near creek, meet, and the interior of the marsh. Okay, so on the right, so this the water fluxes, pore water fluxes, and the down bottom is the DIC fluxes, which is derived from the 1D vertical model, combine the ratios of the isotopes and the DIC profiles. Okay, and then we only did one site in this case for the DIC fluxes, but there's some issue with the sampling. So on the right is kind of the major findings. I'll just go through it. So subsurface sediment horizon intersected by the mean low tide supplies a majority, over majority of the total export DIC. And the DIC fluxes can vary by a factor of 4 between drainage basins, even within the same estuary. Okay, and then 50% of the DIC is derived from near the bank, which is shown clearly in the plot. Okay, so I think I covered this also. And then you can also use radium isotope mass balance to derive the lateral fluxes. And in this case, we use the ratio of radium 224 and 226. And then there's basically 3 end members, right? In this case, groundwater, there's seawater, then there's marsh pore water. So then by constructing the mass balance and the measured activities of the 2 isotopes, you can derive the add member fraction in your tidal water, like this on the right, upper right. And then we also measure the DAC concentration in the, along with the radium, the two radio isotopes, and then the relationship between the DAC and that ratio can be used to derive the DAC flux from pore water exchange. All right, so here's the result. 3 years, one is recent, the other 2 is early years. So 3 seasons, then a couple of main points here. That the lower numbers are PEX is the pore water DAC fluxes, basically derived from the pore water exchange. And then there's a submarine groundwater. So you can see those annual values are relatively small. Even the seawater, groundwater is relatively a small portion of the DAC lateral flux. So the sea level, which is on the blue line, plays a very important role here. So you can see that the higher the sea level, the more of the exchange and the more of the DAC export. And there's a large interannual variability of the signal. And then I should mention that this little bar here, this we only in the fall 2019 we only sampled once in have a sampled one tidal cycle. So that number tends to kind of small. We don't know what's going on with that. So now this is kind of the first time we'll be able to compare all the methods we use for the same, at the same place, at the same time, which is basically the 3 kind of box I circled here. So the MLR tidal model agree reasonably well with the core method, the radium thorium core method, right? So the radium mixing model seems to be, except the one case in the fall, it seems to be have higher value than the other 2. But we're happy because they emerge somewhat, but obviously there's more to be done, the uncertainty of that. So, and I should also mention this is the MLR has much higher resolution than the other 2 methods, which is basically seasonal and over tidal, a few tidal cycles. And to summarize all of that, so DAC export not only varies temporally, very spatially as well, and then there could be a huge spatial variability in one system. Majority of the lateral DAC exports from low marsh sediment horizons intersect with the mean low tide. Obviously, it's driven by the tidal pumping. The radium thorium DAC fluxes agree reasonably well with the time series approach. The isotopic radium balance estimate tend to be a little bit higher than the 2 other methods, at least based on what we have here. Sea level may play a very important role. So this raises another question. When further sea level rise, whether that can drive more lateral export from wetlands, right? So, and then the last point, the different methods may capture different temporal spatial scales. And this is kind of that point. I don't expect you to read all of it, but the highlights, the time series kind of instantaneous, and then the core method integrate over a week or 10 days, and then the radium is tidal cycles, and then this is the limitation obviously for each method. But at least for now, we can think at least for Tzortziou Pond marsh, we think at least the several methods kind of converge on the same reasonable numbers. Okay, so last a few minutes, I'm gonna go quickly. The recent study on CO2 fluxes from Tzortziou Pond, the same site. And then the goal of it is to assess the CO2 effluxes from the tidal creek and inundated marsh water from the tidal to seasonal scales. And then we're gonna separate different sources of which part of the CO2 efflux is actually supported by marsh versus estuary carbon. And so And remember the indented area is changing minutes to minutes, right? So when tide comes in, it will gradually occupy the tide. So we take that into consideration. We're gonna use high resolution PCO2 measurements at the mouth, and then the model to simulate the inundated area, and then we'll combine the two to estimate the total CO2 flux from water. And then we normalize it to the total marsh area, okay? We also estimate marsh contributed CO2 fluxes versus the estuary. So we define the estuary end member, and then we can estimate how much CO2 being added to the estuary carbon, and then calculate the marsh carbon contributed fluxes. And then we've also separate the photosynthesis effects and the respiration over daily scale. So this is kind of the three main analysis. And I'll quickly go through this. So that on the left is basically 2 years, summer to fall deployment of CO2, PCO2 in situ measurement at the mouth of the creek. And then this is a kind of enlarged time series. And then this is the principal component analysis, the lower left. And basically there are two kind of major components control the fluxes. One seems to be the daily cycle of photosynthesis respiration, and the other seems to be the tidal pumping, right? So the upper left, this is the kind of summary of the marsh contributed CO2 fluxes, okay? The upper is the daily estimates, and then I want you to focus on the lower table. So especially, so the first column on the left is the estuary contributed CO2 fluxes, and then the middle marsh, and then the total. So you can see the marsh contribute more than 60 or 50% of the CO2 fluxes, which is not surprising given there's a lot of marsh carbon around. And it's a little bit of a surprise to me, photosynthesis actually contribute quite a bit of reducing the CO2 efflux on a daily basis. And then this is kind of a summary of that. So the photosynthesis effect is left, and the respiration, and then the net total on the right. And then this is the fraction of the photosynthesis on the right, how much of this photosynthesis reduce the respiration rate. So that negative number is just, decrease CO2 fluxes from the respiration. So that's quite a significant, 20 to 40%, right? So, and this is raises another interesting point, which is during this transport, there's some of the DAC actually being converted to DOC and organic carbon. So this is kind of the summary of the two export number. This is just the comparison of the different marsh area rates. Okay, so gram carbon per meter square marsh, and then the DAC export is from this study, and then the CO2 fluxes as well. DOC and POC are from literature. Okay, and then this is kind of the representative of the East Coast. So, but only in the summer and fall. Okay, so you can see this term, this photosynthesis reduction term, is actually quite significant. That's almost the same as the net CO2 fluxes from outgas from the tidal water. Okay, within that, 96 outgas of CO2, 63% is supported by marsh carbon, and then 36 by the estuary carbon. Okay, and then this photosynthesis reduction of CO2 is almost the same as the net outgassing rate, and it's actually quite compared to DOC and the POC flux on an area basis, it's quite significant. So I'm wondering, this DOC flux, is that a lot of that maybe coming from this photosynthesis happening on a tidal cycle scale? So that's kind of or just think about this last night. To summarize, so CO2 effluxes from marsh tidal water are highly variable. Okay, and important carbon loss term in the marsh carbon cycle. It's equivalent to about 20% of the lateral DIC fluxes, and similar to the same magnitude of DOC. On average, 63% of the CO2 flux is supported by the marsh carbon, and the rest by the estuary carbon. Photosynthesis seems to be very important. So it's a significant process converting marsh DIC and organic carbon. So the last, I already said that the question, what kind of role it play in this, lateral transport? With that, I'll acknowledge my funding agency and the collaborators and lab staff and students. Thank you for your attention.
We have 2 minutes for a couple questions.
Hello, thank you. This was very interesting. I was wondering if you could talk a little bit on your model when you were incorporating vegetation, whether depending the type of vegetation, like if you have a more kind of flexible, whether that might impact wave attenuation or like the overall hydrodynamics. And if in that case also, could we potentially use your model for other systems like mangroves and
so Kevin probably going to talk a little bit more, but so for that, the hydrodynamic model, I don't think we have the marsh plants and other feature in there. It's kind of just a water budget model, and it has a morphology into it. The elevation of and the water flow, and so the basically model projects how much water going through the tidal channel versus how much from the side, which is overland. So we only gauge or only measure the the signal at the tidal creek. We don't measure at the side, right? So we use the hydrodynamic model to actually correct how much of the water actually going into the marsh, and we assume those water are the same as going through the tidal creek. So we don't really use the biology of the marsh, but that's a good point. I think we should. Yeah, Galen.
Yeah, thanks, Aleck. I wanted to ask a big picture. How do you see the link from your talk to the talk that Ray Major gave, which is like this global synthesis of everything in terms of the total, all these cycles, bringing together lots of data sets like yours? Like, how do you scale up what you're doing to improve the numbers in that kind of global estimate? Is there a clear linkage there, or what can you comment?
That's a great question, and that's I hope Kevin can answer some of that. but sorry to put you. But we are starting to look at the mechanical drivers. Okay, so in this case, things like water elevation, biology, the morphology of the marsh, and then seasonal and the tidal cycle. So those are kind of few major drivers for this kind of fluxes. We hope that we can have a mechanical model, not the empirical model we're talking about here, to actually simulate those fluxes. I think that's a critical next step for this lateral fluxes. Then we can actually scale, say, the East Coast without just using, we've been using an average for decades, right? So I hope we can do a little bit better than that. Then I think raise, that will come into play, right? So then whether we can scale up to the global ocean, I think that's a little bit of hope for me.
Okay, excellent. My head is swimming with questions already following those two talks. So I'm going to dig a little bit deeper into some of the data that Aleck already talked about. We have now just over 4 years of near continuous data on fluxes at this site at the White Bay National Estuarine Research Reserve to see if we can get at some of the mechanisms and make those predictions that were just talked about there at the end of Aleck's talk. So I want to acknowledge my co-authors here, and particularly Sophie Kuhl and Megan Eagle. The three of us have been digging into this data pretty vigorously for several months now. So I'm at the U.S. Geological Survey here in Woods Hole. We're on the Quissit campus of WHOI. Been there since 1960 something, and I haven't, but the center has. And so this is our team here at USGS working on these coastal carbon topics, and we've been studying coastal carbon here within our group since 2008. We've been focusing specifically on blue carbon aspects since 2011. So just after the term was coined in 2009, as Isaac mentioned. So this is applied science, a lot of end user engagement, working towards implementation of carbon management, and a lot of focus on supporting policy. I think we should all be really clear that pretty much all carbon science at this point is applied science, right? We have some big problems to solve around carbon. So we have a national focus, USGS more broadly across the various mission areas, water, ecosystems, and some others. Also study coastal carbon and coastal blue carbon in particular. So several other groups have been measuring lateral flux at some additional sites in similar ways. So the information is building. So a little bit of context around blue carbon, carbon management, and specifically these lateral fluxes. So everybody knows this diagram really well. Here we have current CO2 emissions, greenhouse gas emissions, and a projection of possible reductions across the century. Here we have agriculture, forestry, and other land uses as carbon dioxide removal technologies. So I've heard these described as granular, meaning as opposed to large geoengineering type approaches for removing carbon dioxide from the atmosphere. We're talking about thousands and thousands of different projects at the local scale that we hope can accumulate to a significant removal of greenhouse gases. One key feature of them is that we have the technology for these kinds of natural climate solutions now. And so the hope is that we can turn this emission into a sink relatively soon while working on more industrial types of carbon dioxide removal. There is some evidence that this is working. So this is an organization called VERA. They have something called Verified Carbon Standard. This is a voluntary carbon market methodology. So there is, for several years now, there's been a methodology for how to account for carbon dioxide or methane removals from the atmosphere due to blue carbon projects, restoration, et cetera. And within the carbon dioxide removal world, people talk about gigatons. They mostly just want to know about what can remove gigatons per year or more. So already this one organization is identifying about a gigaton of carbon dioxide removed over the last several years. And this is very early days. So I think these granular approaches can add up. So global blue carbon potential is, it's not massive compared to gigaton type removal approaches, but I would suggest that the potential is far from fully understood. But it probably is approaching the gigaton per year carbon dioxide removal equivalent. And in order to properly account for greenhouse gas removal potential, we have to consider avoided wetland destruction, restoration of degraded wetlands, avoidable methane emissions, and contributions to ocean alkalinity and long-lived dissolved carbon pools in the ocean, and enhanced future emissions and losses driven by sea level rise and coastal protection. Pretty much none of that's been done yet. So there's a lot of work still to be done, but I think at present we're underestimating the potential. So just a little bit of other work that we're doing around coastal carbon and blue carbon. I've identified these kinds of settings here, diked and impounded former salt marshes is an important source of methane to the atmosphere. And so if we manage those more effectively, allow tidal exchange and salt water into these systems, we can reduce methane emissions significantly. One problem with doing this, though, is that we don't have these kinds of settings, drained wetlands, impounded wetlands, tidally restricted wetlands, well mapped. These red lines here indicate road crossings across the tidal space. And these are dikes here. So we have quite a lot of managed wetland that has a lot of restoration potential and greenhouse gas emission reduction potential. But these are not properly mapped at this point. So we're working on those types of issues as well. So far, we're finding that the existing mapped databases for the United States on wetlands underestimate tidally restricted and impounded wetlands by about a factor of 2. This was work by Jim Holmquist at the Smithsonian. And my mouth is like paper. Is there any water around? So we're also working with folks at the University of Connecticut, Tzortziou and colleagues to develop remote sensing products to automatically detect these kinds of changes in the ecosystems and characterize managed condition as well with rapid updating with Landsat imagery. So accounting for the lateral flux in blue carbon. To make lateral flux component of blue carbon operational as a natural climate solution in carbon markets and national greenhouse gas inventories, we need to be able to measure the flux reliably and with known uncertainty. We need to know the primary controls and be able to model or otherwise predict fluxes and change in fluxes with both global change processes and management actions. And we need to understand long-term storage in the ocean. So as Aleck mentioned, we made an attempt previously in 2018 to pull together scientists collecting this type of data and synthesize the existing data and see if we can come up with improved estimates of flux and identify drivers of flux. It was a very effective discussion. We came to agreement on what the key approaches were and interpretations, what the best ways are to measure the flux. But effectively, there wasn't sufficient high-quality data to support a new synthesis. We're trying to work beyond what we did previously in a Najar et al. Led study supported by NASA to synthesize the existing data. And we clearly showed that tidal wetland lateral flux is a pretty substantial part of the coastal carbon budget, but these are based on existing literature with basically old school techniques, which involved measuring fluxes across individual tidal cycles. And I'll talk about why that is efficient. So I'm going to present extended time series data at a single site to see if we can achieve some of these objectives. And the hypotheses are that lateral flux is related to the rate of carbon cycling within the wetland, so things like gross primary production or respiration, and that it's related to the rate of aquatic exchange between the wetland and the estuary. So this data set is 50 months of data. We have about 43% coverage across that time frame. So it's likely the longest high-frequency data set available. And the goal is to extract the fullest knowledge that we can from the data. So Aleck already presented several papers from this data set, but we've expanded the length of it, and we've gone back to the beginning, and we're analyzing 4 drivers. Also, the dissolved organic carbon portion of this data set hasn't been. Oh, thanks. Yeah, I really appreciate that. I think I was mouth breathing inside my mask. So the DOC data set hasn't been published yet as well. So that portion is new. So we used a similar approach to what Aleck described, a statistical model to predict dissolved organic carbon concentrations at the mouth of the tidal creek over time. And so this is predicted versus measured DOC concentrations. This is our model. We're predicting based on temperature, fluorescent dissolved organic matter, and pH. When the FDOM sensor wasn't working, we were able to predict reasonably effectively with just temperature and pH. So although there's some uncertainty in the predictions of concentration, the range of water fluxes is much larger, so that the fluxes are actually much more closely aligned with between measured and modeled. So this is the data set. It's 186,000 observations of dissolved organic carbon and inorganic carbon at the mouth of this creek across 50 months, as I mentioned. So these are all instantaneous flux, grams per second, exchanging between the estuary and the wetland. Right away, we can see that the elevation of the marsh relative to the tide is important, which is one of the hypotheses. So I'm going to very quickly, in the next couple of slides, go through some of the data analysis that we've done. Some of this is new. So beyond what Aleck just presented. So the sensor deployments and maintenance, YSI, XO2, and a Sontec IQ acoustic flow meter, creek sample collections across a number of cycles, periodic measurements of terrestrial, freshwater, groundwater discharge, which does carry its own carbon source that we need to subtract from the total measured fluxes. And a lot of sensor hygiene, data hygiene. So a lot of the data either needs to be corrected or discarded if it goes outside of bounds. And quite a lot of ancillary data, including elevation, high resolution elevation on the platform. We updated the overland flow predictions. So as Aleck mentioned, a lot of the flow goes through the creek, but when the tide is above the platform elevation, then you have sheet flow onto the control space. And so that's not accounted for by the sensor. So we have an oceanographic model for predicting those fluxes, but we extended the range of those predictions to higher tide levels. And so it's a little bit different than what we did previously in terms of those corrections. So we adjusted the ebb flows to close the water budget to net freshwater outflow seasonally. And the total net freshwater outflow is calculated by the isohaline method from McCready. And that includes evapotranspiration, rainfall measurements, and groundwater flow by difference from these three. And we have a slightly different DIC equation, provides identical results to the one Aleck showed. And we limited the carbon concentration calculations to the ranges of our sample collections, conditions during sample collections. And I did a bit of gap filling. Very importantly here, we found that a number of the tides had highly unbalanced flows. And so what we're looking at with each of these tides is net of flow in and flow out. and it's a very small difference between those two. If the water budget is incorrect, then you get a wildly large flux, either inward or outward. So we had to do a lot of removals of those large tides and adjustment of the total flux based on the frequency of large tides and the average flux during those tide heights. We adjusted all of the flows. We identified a bias between observations of flux data and observations of flow data. We had a lot more flow data than we do flux data, and so we adjusted based on the frequencies of the two, and did a lot of sensitivity analysis to estimate uncertainties. And so the point is that these are not just simple measurements, collection of data at the mouth of a creek and getting the flow. There's a lot of work that has to be done to identify biases and errors. And just to give a visual for one of those, here we have the whole data set, DOC fluxes, and the black shows flux based on concentration and measurement of flow in the creek. And these other colors here indicate after corrections for overland flow, for balancing of the flow budget seasonally, and the groundwater correction. And we end up with this kind of data, very obvious spring neat cycles, for instance. And these are the outcomes in terms of time-integrated fluxes. So across 4 years, the DOC flux ranges from 60 to 130 grams of carbon per meter squared per year. The DIC flux ranges from 175 to 403. This is part of the data. This 403 is part of the data that Aleck presented, but it turns out interannual variability is substantial. And seasonal cycles are really obvious. So the summer here is in this magenta color, I guess. And so they're not fully consistent. We have here a fall season. This is the highest fluxes of all. So DOC exports relative to DIC exports across these seasons and years, seasonal in black and annual in red. So the seasonal range of the ratio of DOC to DIC is 23 to 55%. There's a tendency towards a higher ratio in the summer. Annually, the range is much tighter, 32 to 34%. So the question is, why so consistent? And I can tell you that initially it wasn't like this at all. It was after many efforts at correcting all of the water budgets that this pattern fell out. And but I think the basic answer is that the main drivers, the source, plant productivity and respiration, and the hydrodynamics and hydrology are consistent across all of these time frames, of course. So here's a first look at the effect of platform elevation, which was one of our hypotheses relative to sea level. Here we have high tide level for each of all of the tides in the data set, and we have DOC export in the black circles, and we have the percent per each tide height of the flux and of the number of tides. What you can see is that when the platform hasn't been inundated, the flux is very small. 80% of the flux occurs during kind of standard normal tides that flood a portion of the platform. And when we get up to the highest tides, which include storms, individually those tides do have a larger flux, but they're only responsible for about 10% of the flux across years. DIC has the same pattern. We have this import of carbon that occurs at this particular tide height, and meant to mention, we have some ponds in the platform that appear to retain water. So we have generally at low tide levels, we have net drainage of water. Higher tide levels, we have net import of water. So there's a temporary storage. So what's the expected influence of carbon source processes? So productivity and respiration rates should have a clear influence. Growing season productivity is generally, in the literature, is generally closely related to photosynthetically active radiation. Ecosystem respiration closely related to air soil temperature. This is a paper that we have in review led by Joe Carey at Babson College. Very clear relationship between ecosystem respiration measured as a CO2 flux from the platform to air temperature in this case. So we would expect higher DIC fluxes as that respiration source increases. But something that's very important to note is that in any of these tidal wetlands, the water table is going to vary in height, right, over time. And we have a lot of autotrophic and heterotrophic respiration, particularly in the rooting zone here. Some of the time, that respiration is going to be happening above the water table, and therefore it'll contribute to a flux to the atmosphere. Some of the time, it's going to happen below the water table, and therefore it'll tend to contribute to the lateral flux. And so what I'm pointing out here is that we don't actually know what the respiration rate is. We don't know what the gross primary production rate is, because there's always this unknown portion that's happening below the water table and contributing to this flux. So these are definitely underestimates of the rate of respiration. But generally, if we look at time integrated fluxes, so these are seasons across the 4 years, we look at DIC flux versus air temperature, there's a pretty good relationship, versus mean sea level, pretty good relationship also relative to tide range. This one point, which is fall of 2014, clearly falls off this source driver relationship, but when we look at it relative to the driver of water exchange, it's not an outlier anymore. We have pretty much the same relationship with DOC. If we look at a slightly shorter time frame at 2-week intervals to give us more statistical power, and I'll note that these 2-week intervals of data have water budgets that are closed based on knowledge of the water budget across the whole season. We can get a pretty good prediction of measured DOC flux based on tide range, air temperature, and average sea level. PAR was non-significant, very poor relationship. We don't do quite as well with DIC, but still a significant relationship relative to air temperature and average sea level. Tide range didn't show up as significant. So I think there's a lot of progress here towards being able to predict these fluxes. These drivers are features that we can map. So here in the United States, we have now new maps nationally of elevation of salt marsh or tidal wetland platforms relative to high tide. And there are products, one from Rusty Fegan for gross primary production, and a new one that's coming together from Victoria Wolf, the USGS, NPP for tidal wetlands in the United States. So we have the drivers, we have pretty good relationships with the flux. I think we can start to improve on our estimates of tidal exchange. And so Z-star is an indication of elevation of the platform relative to high tides and mean sea level. So here we have a pretty good relationship seasonally, although that should be a lowercase Z-star because Z-star is relative to tidal datums. So hypotheses and testing predictors, results support the notion that we can predict these fluxes based on hydrogeomorphic setting and productivity. High frequency data is required to capture some of this temporal variability. or at least extended data, I should say. Substantial corrections to the water budget are necessary. And I'll note that if anyone has data like this to contribute, we'll make another effort to synthesize data. We have a Mendenhall postdoctoral fellowship that will be available in the coming months. We have an AGU session on coastal carbon cycling, and these are our sources of support, and thanks to the WHOI Bay staff. Thank you.
Thanks, Kevin. It's Ray Najar from Penn State. So I thought I heard two different messages from you and Alex. Alex seemed to be emphasizing the importance of like these extreme events for dominating the export. And I thought you were saying, not that they were trivial, but a lot of the action was kind of in kind of regular tides. So I'm probably just not understanding something, or
no, I've noticed that we've said different things about that. So, you know, obviously there are examples of storms that drive a significant flux, and I think that's probably what Aleck is responding to. But when we integrate over time like this, they just, on an annual basis, basis, they don't turn out to be a substantial part of the flux.
Right.
But that is the least well-known part of the flux, by the way, though, because when the water levels are very high, understanding the water flows is much harder.
You have to rely on that hydrodynamic model. And
yeah.
Right. Got it. Thank you.
Just a quick comment to that. The data I show, basically several months. What the Kevin shows, several years. That's a difference. So you can imagine at a given month, storm, and then the water budget is just basically your fluxes. That could happen.
Hi, Debbie Steinberg, Virginia Institute of Marine Science. I'm wondering what role, if any, the movement of organisms might play in lateral exchange, like larval fishes, say, feeding and then moving offshore, or if that's even been looked at all, because there's definitely movement of organisms with the tides. There's also the seasonal movements of fishes that are using these areas as nursing grounds. And has anybody looked at carbon transport that way?
Yeah, it's a good question, and I don't remember the answer. I believe it was looked at in a getting the author's name. There was an examination of that, whether it was a significant flux. It didn't seem to be a really large flux, but more importantly, you wouldn't expect that carbon to reside for extended time. So it probably is part of the budget, but not part of blue carbon for sure. That makes sense?
Hi, Kevin. Really nice talk. Amanda Spivak at University of Georgia. Maybe this is for both you and Alex. So there's a lot of focus on de-icing as CO2 and bicarbonate. But have either of you looked at the contribution from methane?
Yeah, good question. So Rebecca Sanders-Dumont, a postdoc that's just wrapping up her time with us, did quite a bit of methane flux measurements there at the site, though they were towards the landward end of the platform, towards where there's a little bit of freshwater influence from groundwater, so looking at some Phragmites locations. They're significant in terms of, in that lower salinity range, they're significant in terms of the radiative forcing, but not in terms of the carbon budget. There are a lot of methane fluxes and flux measurements as well from Serena Mosman-Valtierra at University of Rhode Island, and some folks in Jim Tang's group, based on chambers on the platform, and they tend to be very low in the main platform here where the salinity is high because of sulfate inhibition. There are some methane fluxes in the tidal water exchange as well, which might be part of what you're asking about, I just realized. So yeah, there's a very obvious signal of higher methane concentrations in the tidal water at low tide, probably where there's some influence from freshening due to the groundwater.
No? Okay. Oh, here it is. Okay, thanks. Okay, great. Yeah, I'm Patty Oikawa. I'm from Cal State East Bay. And I'll be talking today, inching more and more towards the atmospheric side. Kevin's work really lines up well with this. So combining atmospheric and lateral carbon flux measurements in tidal wetlands, and mostly focused today's talk on San Francisco Delta region. I have a lot of collaborators on this talk, and I'll be calling them out. So this is already, a lot of this has been said, but dating back to Odom's outwelling hypothesis, we know that this is a very significant flux, but like the previous speakers pointed out, we have very few studies that are doing continuous data collection of lateral carbon fluxes over multiple years, and even fewer who have combined these with the atmospheric components. And so that's what I'll be focusing on today. And then also drawing attention again to the chemical composition of this laterally transported carbon is really important for blue carbon initiatives, like Isaac mentioned earlier, and that we need, advocating for the need for all these different types of flux measurements to get at net ecosystem carbon balance, which is something I'll come back to a few times in the talk today. And so really we're, again, just focusing on the CO2 and methane fluxes that are exchanged atmospherically by the ecosystem, combining that with these continuous flux data transported laterally, and then also using sediment information to inform carbon budgets. So my background is mostly from the atmospheric side of things. So I'm an eddy covariance expert. And for those of you who are not familiar with eddy covariance, this is where we have long-term deployment in situ sensors shown up here on the left that are infrared gas analyzers. They measure concentrations of methane and water vapor. And we pair those data with sonic anemometer wind velocity data in 3 directions. These are super high frequency, 20 hertz data collection. And they give you integrated fluxes across the land surface every half hour. So if you look at this example wetland here, this is your eddy covariance tower. For every 30 minutes, you get an integrated flux of net exchange of CO2 between the atmosphere and the ecosystem, or methane or water, whatever you're looking at. What Julie Shahan did in my lab, she has a paper coming out soon, is also kind of taking this rich data and decomposing it, or we call it partitioning it, across your different heterogeneous land surface. So in this example, we have quite a bit of mudflat in the center of our footprint here and mixed with vegetation. So what she did was decompose the flux data and look at every half hour, and if it's dominated by vegetation versus mudflats,
and pulling out the ecosystem fluxes depending on those footprints. So for the top panel here, we're looking at net exchange of CO2, in this case, micromoles per meter squared per second. Negative numbers mean uptake of CO2 into the ecosystem over an average day here. And you can really see the vegetated dominated footprints remove a lot more CO2 relative to the mudflats, although even the mudflats remove a little bit. And then for the methane signal, in this case, this is a polyhaline tidal wetland, so there's very little methane emissions in either case. Oh, sorry. Okay, so then for our lateral carbon fluxes that we're measuring in our group is really similar to Aleck's approach and Kevin's approach, where we have an ADCP installed in the tidal channel, and we do some channel rating to help us estimate water velocity and fluxes across the entire channel. And then we have a YSI sonde out there collecting extra data proxies for the POC, DOC, and then we have a PCO2 sensor as a proxy for DIC. And so then we do these bottle samplings quarterly across tidal cycles, where we do some Van Dorn sampling up there and send those off to be analyzed so we can get an estimate of total alkalinity and DIC, POC, et cetera. So this approach is really informed by my colleague, Brian Bergamaschi, who's at the USGS and has published this approach previously. But this is something that we're really working on in my lab right now, is kind of doing a little bit more of like what Kevin did, is a lot of diving into the details of this. But basically, at its essence, is you're just taking your discharge time, your concentrations of whatever species you're looking at, and then dividing it by the marsh area. But there's a lot of nuance in this. And what we're now doing is working on specific code to help us better estimate, in particular, the marsh area, how much is flooded at every tidal cycle, how much water is entrained and kept in the pore water, et cetera. And then this figure is just showing that the time series of some of these variables. Okay, so for the majority of the talk, I will be focusing on two sites, the Rush Ranch site up here. So this is San Francisco Bay. There's the Golden Gate Bridge, for those of you who aren't familiar. But Rush Ranch is up here. It's at sort of the entrance of the Sacramento Delta, which is pouring tons of fresh water out here into the bay. So we've got this nice salinity gradient of completely fresh to saline. So this is a brackish marsh complex. And then Eden Landing, our other site, is a polyhaline, recently restored tidal marsh. But this is a very important estuary to the West Coast. Lots of wetlands. Like I said, nice salinity gradient for studying these processes. It's also highly urbanized. Disturbance is a big factor here. It's also, wetland restoration is a really hot topic in California right now. They recently, California released its scoping plan, which is, sort of our blueprint for how to get to net carbon neutrality by 2045. I hope we can do it. A little incredulous, but so wetland restoration is one of those tools in the tool belt that the government's hoping to help us, reach this goal. They've identified an additional 50,000 acres for potential tidal wetland restoration. So there's a lot of policymakers and a lot of neat applications of this kind of data, and people interested in blue carbon potential. Okay, so what I'll do is introduce you to Rush Ranch first, the site and some of the data, and then go on to Eden Landing, and then I'll end with sort of some of the larger projects that are going on and more national scale type work. So for Rush Ranch, we have, again, it's a brackish marsh. It's historic, so it has very low disturbance. It has a pretty low but variable salinity range. It's located within a NER, so we've got great historic long-term water quality data. This is a photo here of the eddy covariance tower that was set up by Lisa Marie Wyndham Myers in 2014. And I think it is the longest running eddy tower in a tidal wetland in the US. I'm not totally sure, I think so, and maybe in the world. And then they installed their lateral flux station in 2018. So they also have multiple years of data, maybe not as much as you guys, but pretty close. And okay, so this is Matt Bogard's paper came out in 2020, which was the first paper to integrate the atmospheric with the lateral. And he was working with 1 year of data. So in that study, they found that atmospheric removal, so this is what you get from the eddy covariance tower, was around 250 grams of carbon per meter squared per year. And the lateral export, which they were able to account for DIC, DOC, and POC, was about 100. So it's about half that. So we're looking at lateral fluxes at a scale of 50% of the atmospheric side. Again, going back to ODMs, that's fitting in with that hypothesis and a lot of the other data that have come out since then. And so, like I said, we have multiple years of, we got a grant to support extra years of data. We're still crunching those numbers, unfortunately, but we do have more of the eddy tower data crunched. So this is work coming out of Sarah Knox's lab. Sarah Russell is the PhD student leading this effort. And so this top panel is showing cumulative NEE, so net exchange of CO2 cumulatively over multiple years. So we have, this is our 5th year, it's a little short, but you can see there's really high interannual variation in productivity at this site. So this 1 year, this red year here, is actually showing a net source of CO2 to the atmosphere from the wetland, which is really unusual. Contrasting with this blue year here, which had a net uptake of around 400 grams of carbon per meter squared per year, which is really high. So that's sort of on the higher range you would see in a tidal wetland. And what Sarah's analysis has indicated is that a lot of this might be driven by salinity changes in the brackish marsh. So we had pretty different salinity across these years, which is a function of, you know, drought years and wet years in California affecting that. So I think it's going to be, I think, you know, one of the power of bringing these two types of data sets together will really be highlighted when we get the lateral flux data fully analyzed and can look at this interannual variability and how interannual variability in your primary productivity then cascades down and affects the lateral fluxes. So I'll come back to that again later. Okay, so the second site is Eden Landing, and this is the site that my lab runs. Actually, really quickly, this is part of the South Bay Salt Pond Restoration Project, the largest tidal wetland restoration project on the West Coast. So we used to have a lot of these giant salt ponds in South San Francisco Bay. This is what they used to look like, and they've done a fantastic job restoring those. So this is the whole, you know, part of this complex here. And so it's a very diverse complex with, it's highly managed, highly disturbed. There's no natural inflow of water or sediment from like a creek, like you guys have here in the East Coast. So we just have kind of runoff, surface runoff from highly urbanized environments, and then, but most of the water is just coming in from the bay and being exchanged with the bay. And our eddy covariance tower here is installed in Mount Eden Creek Marsh, which was restored in 2008, so it's pretty recently restored system. Again, it has a lot of bare mudflats and some vegetation, so it's actively restoring. So the vegetation is really growing and expanding every year, and it's a sort of a dynamic setting. Our tower was installed in 2018. Our lateral flux station was installed in 2020. Again, it's super polyhaline, high salinity, low elevation, and high tidal range. So it's a very different site than Reshiram. Okay, so the first I'm going to start with the atmospheric story. And from these 3 years that I'm presenting here, it's again very different from Resh Ranch. We have very low interannual variability in productivity at the site. So we're very consistent around 400 grams removed. So as I mentioned before, that's pretty high for a tidal wetland, and it was pretty surprising to us to see that because, again, we've got these short little stubby pickle weeds and things like that. And so this is a hint to me of lateral fluxes because some of this is actually not getting respired and the CO2 coming out, and we can see it with our tower, it's more probably getting transported. And then the methane story is, again, it's very low, very, very small amounts of methane. Another first hint at the lateral fluxes at this system was a paper that Ariana Arias-Ortiz et al. Published in 2021. She's a postdoc with Dennis Baldacchi at Cal. And she combined the atmospheric data with soil sediment cores. And the data from Eden Landing are in the red box here. And you can see, again, we see with the tower, 400 grams of carbon per meter squared per year removed. But in the sediments, what she found is a soil organic carbon accumulation rate of only 70 grams. Right, so there's a huge, huge difference here between these numbers. And what's also totally fascinating and why this paper is so cool is she did this in a couple of other freshwater peatland sites in the Delta and compared, again, atmospheric data to sediments, and they match almost perfectly. It's so interesting to see these different types of wetlands and the different carbon dynamics. But everything you see atmospherically pulled into that wetland through photosynthesis stays there. Again, there's something going on for Eden Landing. Okay, so we only have 1 year so far of lateral flux data for Eden, and that's what's presented here. And we also only have so far the DIC. So we're just going to focus on the DIC fluxes. The atmospheric data are on the top here, a little bit of a higher removal year than we normally see, around 500 grams. And laterally, it's really interesting, over the entire year, cumulative was only 70. But what I really want to highlight for you all is this growing season data in the red. You can see begins around day of year 100, photosynthesis turns on. You see this really strong drawdown of CO2 into the ecosystem ending around day of year 250. And with the lateral fluxes, it's also when you see really strong negative numbers, meaning export. So we're pumping out a lot of DIC during the growing season associated with productivity in the wetland. And this is also, vast majority of it is in the bicarbonate form. But when you add in an account for these wintertime periods, we actually start growing. And so we start to import DIC during these periods, which is not what I expected to see and hasn't been seen, I don't think, in other wetlands. And so we did some radon sampling to test for the presence of groundwater. There wasn't any evidence of groundwater in the winter or the summer, but it's something we want to revisit. So we started to think about the nutrient story here and just wanted to highlight that there's a lot of nitrogen at this site. So these are data coming out of a NASA grant led by Jim Holmquist, and Erica Kuntz is leading up this paper on pore water sampling that we've been doing across tidal wetland NERs. And you can see she sampled across all of these different sites. The Eden Landing is one of the highest for ammonium in pore water. And this is one of my students collecting pore water. And so we know we have eutrophication problems in San Francisco Bay and algal blooms and things like that. So I was interested if there might be any evidence of, algal bloom activity, and maybe that's a source of DIC in the winter. But it doesn't seem like that's the case. So this is the data. These are all very preliminary, but I'm now showing my cumulative DIC. You guys have already seen this on the top panel. But then I'm including also nitrate fluxes, and this is from a SUNA UV nitrate sensor that, again, Brian Bergamaschi installed at our site. And then we also had measurements of chlorophyll. And so what's really interesting is that we have a lot of import of nitrate and the tiny exports of chlorophyll during this growing season. But in the wintertime, things really die down. There's not a lot of activity, not a lot of changes in our fluxes. We know we have high nitrate, but it doesn't seem like it's explaining this story. So my student, Maya Matsumura, just put in a proposal to do some isotopic measurements and see if we can maybe parse out these different sources and sinks of DIC at different times. Okay, so now I'm going to take a step back and talk about some of these larger scale studies that we're working on. Right now I'm leading a new DOE grant, which is coordinating eddy covariance measurements in tidal wetlands across the US and Canada. Eddy covariance in tidal wetlands is also kind of a young field. I mean, we were talking about lateral fluxes are very, very young. These are just like the toddler version of that. And so this grant is really focused on like coordinating that, creating a network, making sure we're processing data in the right way, partitioning data, et cetera, and also collecting the right kinds of ancillary measurements. So things like phenology are really helpful for connecting with remote sensing, water quality, pore water data. And we're going to be really focused on, with these 7 towers, on things like pulses of nutrients and salinity, on productivity and methane. But we're also going to be very well poised for adding in lateral. So some of these sites have lateral measurements, some of them don't. And so what we'll be doing next is hopefully adding those systems so that we can get at larger scales and encompassing more diversity of wetlands. So if there are any program officers out there, we might be sending you a proposal soon asking for more money. And then also, very, very excitingly, Adina Paytan just got a large grant from UC. And she's going to be kind of doing what I was just talking about, but at the scale of the Bay Area. So I already talked about Rush Ranch and Eden Landing, but there's a couple new sites up here, Dutch Slough and Hill Slough, being run by Dennis Baldacchi's lab, and those are freshwater tidal ecosystems. So those are going to have some really great, compelling data. And then Adina is getting into the eddy community. So she just installed 3 towers at Elkhorn Slough last week. So all of these sites, there's going to be 7 towers, all of them equipped with lateral flux stations, ADCPs, the works. So there is going to be a lot more lateral flux data coming out very soon. Okay, so sort of concluding up here, obviously I'm advocating for combining atmospheric with lateral data as well as soil sediment and pore water data to give us a better view and understanding of the whole picture of carbon cycling in these ecosystems. In our site, you know, we found during the growing season, at least, we were exporting around 200 grams in the form of DIC. But, you know, when you start to get these long-term continuous data sets, you start seeing things that are surprising. I think that's going to be something that comes up more and more. There's a lot more variation out there, a lot of things we don't understand. So again, this is such a young field. But hopefully we'll be growing and learning about these things together. I think some of the big areas to look at are the influence of disturbance on these processes like restoration, eutrophication, salinity, interannual variation. Scaling is something that I haven't talked about that much yet, but as I mentioned before, these eddy covariance towers are only measuring one little part of the wetland at any given moment. And usually when you're measuring these lateral fluxes, it's across a much greater scale. So integrating these two, they're at different spatial scales, so that's going to have growing pains associated with that. Adina Paytan's group, like I said, she's putting up 3 towers in her wetlands, so trying to get better at like the spatial variability of the fluxes. I think we're going to be getting at that, but it's a huge issue. And then again, this is all within the framework of blue carbon, and like Isaac was saying earlier, this is a huge chunk of the pie. We need to better understand it, what species the DIC is in, and how it can be communicated with policymakers and brought into carbon initiatives. With that, I'm just going to thank my sponsors and some of my key collaborators. Yeah, any questions?
Thank you, Peggy. A couple of minutes. Questions?
Martina, Dublin University of Technology, Sydney. I guess in terms of the San Francisco Bay sites you've been looking at, does the restored site give you any early insight into how to restore sites better? You know, you've been there only 12 months, the footprint's pretty small, but yeah, I guess I'm sort of thoughtful about how you gain insight fast from sites where you can do this intensive measurement.
Yeah, I think there's a lot to be learned from these sites. I do think, you know, ideally, would have more. It's such a highly managed system, and it's surrounded by all this urbanized territory, but there's not a lot of room to grow, and I think that's a huge weakness of it. Also, again, another big weakness is the sediment supply from having natural connections with and rivers would be also ideal. So those are huge weaknesses in this, and I was actually speaking with a policymaker yesterday about this, and you know, how do we account for blue carbon potential in wetlands that have places to move and adapt, and those that don't? And should we not even include, right, the ones that don't have place to move, because they will eventually drown out with sea level rise projections for the bay, at least. So, good questions. But I think we are learning. It's such a big project, and we're still learning a lot. So I feel like it's a good place to study.
Thanks. That was great, Patty. Really cool data. So thinking about this question you just talked about, so it's still a managed wetland. The hydrology is managed, and that kind of mysterious import of DIC. I'm just wondering if there's a change during that season in the hydrodynamics. Are the water exchanges entirely managed, or do they flow freely through an opening, that sort of thing?
They do flow freely through an opening, but there are some other managed wetlands around it where they kind of more manage those water tables, and at some points they, release water into the channel or take up water, but that's all downstream of where our lateral is installed, so we're hoping to kind of avoid that from that. But I think it is a good point to raise. We have a really good relationship with the project managers there, and maybe I could ask if that year if there's something weird that was going on. Yeah, it was during COVID, so I don't know. Maybe people weren't showing up for duty or something. Yeah, that's a good point.
Hi, R.A. Chelsky. Is it possible that the regional sand upgrade could be affecting or contributing to some of the interannual variability at the Rush Ranch site?
Sorry, the what?
The regional sand upgrade, the wastewater treatment upgrade that's reducing the amount of nitrogen reaching the system?
Yeah, that's a really good point. They didn't look at, in that case, I don't think they were looking at changes in nitrate or nitrogen availability in the wetland. That's a really good point. It's something that wasn't included in Sarah's analysis, because they didn't have great data for that time period, or part of that time period. So, but it's something that could definitely help explain it.
I'm just, thinking for the production, maybe.
Yeah.
We have an anonymous question online. Can flux towers be deployed, or are they effective in the nearshore zone, for example, above water instead of above vegetation? It'd be interesting to see the CO2 flux in water receiving DIC flux from the wetlands.
I would love to do that. Yeah. No, I think that would be, it has been done before, it can be done. And yeah, I think especially for the seagrass community, I think it could be used. I know I was talking to some people at Davis who were thinking of doing that in Bodega Bay, because Bodega Bay is north of San Francisco Bay, but they have tidal marshes that are adjacent to seagrass, eelgrass meadows. And so I think it would be a great, I would love to be involved with something like that, but you could have eddy covariance towers and DIC measurements in the water and look at how they relate to each other and interact. Yeah.
So the marshes build soils each year, and this allows them to gain vertical elevation of the tidal frame. It allows marshes to keep this rates of sea level rise. But we know that our marshes aren't 100% efficient at keeping all the carbon they capture and photosynthesize in the system, right? We were talking about fluxes. And so what we want to know is what is contributing to these fluxes. Really important part of the carbon contributing to fluxes is rapid turnover of recently produced organic carbon fixed within 24 hours. About 30 to 55% that is returned as inorganic carbon through respiration. Rapid turnover. Another key part is what's going on in the sediment, so the organic carbon. And so if we can think about our soils in this kind of a profile, where we have our highest soil organic carbon concentrations in the surface, and this coincides with the rooting zone, the active rooting zone. And then we can think about our depth axis. And because our soils build accretion each year, we can also think about this as a time axis. Expiration all throughout this profile, and so that's why our soil organic carbon so the material that gets respired then has the potential to be lost in the atmosphere, lateral fluxes. Carbon in each of these horizons, fastest rate, and as we go down core, we have slower and slower. What happens is when we have this really fast turnover on the order of, well, even hours to days to decades, we have a winnowing of the organic matter. And so a very small fraction actually survives transit through the rooting zone. And at depth, we've accumulated organic matter that's turning over very slowly, so centuries to millennia. And this isn't necessarily a function of the organic matter itself, but the environmental controls on microbes at these depths. So what are they, what's allowed, what's available to them to access? And we have limited loss of this organic matter over time. And this slow cycling pool that accumulates, this is actually the really critical pool that we want to think about in terms of marshes staying where they are in the tidal frame and continuing to build elevation, driving sea level rise. But relevant to this session is we want to think about what's being lost throughout this soil column and how that material is how it's contributing to vertical and lateral fluxes. We've been thinking about this a lot, and with colleagues a few years ago, including Liz Canuel, who's back there, we came up with several factors and mechanisms that we think are particularly important. One of those is the plant species litter biochemistry and rise of deposition. Another is redox oscillations, so driven by tides and bioturbation. Another is these root microbiome interactions. Another is mineral microorganic matter interactions. Now, these are really big topics, but what I'm going to do is tell you a few vignettes, probably too many vignettes, about some of our recent work done to address our research questions. How do litter quality and the environment that is deposited in affect bacterial assimilation of marsh grass carbon? How do redox conditions and root exudates interact to affect the soil metabolism and the mobilization of mineral-associated organic matter? And so this is really focusing below the rooting zone, so the zone that we think is more stable over a long time period. What are the effects of decadal disturbances on soil organic carbon composition? Lastly, we want to think about what are the turnover rates of soil organic carbon under natural and undisturbed conditions. So, after thinking about detritus quality, and our conceptual model here is that marsh grass detritus of different qualities is deposited. Our Pac-Man microbes decompose that. Some fraction is then released in inorganic compounds, and some fraction is then exported from the system. Other fractions are taken up by our very happy benthic who themselves turn over, and the organic matter, either that they're releasing or through their neck, the bioavailable carbon or bacteria. And so what we wanted to test in this experiment was how does bacterial assimilation of marsh grass detritus depend on the availability in the system, quality of that detritus, and then the presence of. We first started off by leveraging a long from these this tree are gauged against this reference low and ambient nature. What we did is we collected cores from these tidal creeks, and then to some of those cores we added labeled 13C sodium bicarbonate, and so this would be taken up by our benthic microalgae. And to other cores, we added 13C labels for Spartina alterniflora detritus. And so what we're looking at here is the incorporation of 13C from the sediment pool into two classes of lipids, fatty acids, and these are so in these top two plots, we're looking at our sodium bicarbonate. That sodium bicarbonate has been taken up by benthic microalgae and somehow transferred to our bacteria group. Variability in uptake rates are
nutrient availability. So our Spartina alternative flora experiments, we just did these in August. Again, a really similar story. We see uptake of the 13C from the Spartina into, and that happens fairly. The other thing that I wanted to point out is that the percent 13C incorporated by the bacteria is similar whether or not the bacteria are getting that carbon from benthic microalgae or Spartina. We're able to access what we think is being lower quality material despite of nitrogen availability. This kind of stumped us for a little bit, and we're like, well, maybe the problem is that we just use them fresh detritus. So we conducted a second experiment where we provided tidal creek sediments, this time from Sage Lot Pond, which is just down the street, with either fresh or leached marsh grass detritus that we created by adding a 13C and 15M. Sorry.
They're not hearing you.
On my shirt. Provide them with different kinds of detritus, fresh or leached, and we also wanted to know whether or not the what we're looking at here. So one of the first things we polyunsaturated fatty acids. When they're alternative to fluorocarbon, if next thing we wanted to know was whether detrital quality and the presence of benthic microalgae affected the retention of carbon and nitrogen in the system. And what we found is that when benthic the take-home messages from this section of their detrital. That rise of deposition, oxygen, and bioavailable compounds stimulate decomposition, mobilize mineral-associated organic
manipulated redox conditions by how the response 1 and 5-day treatments, but I'm that take advantage of new carbon inputs. The other thing you've probably noticed staring at this, that it appears that we have higher DIC production rates for anoxic treatments. And so this is unexpected. It seems always fun. After thinking long, bioavailable compounds. All of our treatments. So there are a couple. May not be accessible to. Our take homes from this section. That microbes below the rooting zone respond quickly to their input. I don't quite understand. We don't have evidence that adding we've been talking about. We've been using the plant.
on just what we wanted to know. Microbes actually eat. Oh, sorry. All the issues out this morning. Is that the soil bacteria are decomposing mix of carbon sources. Now we can do a little thought exercise about the role of decomposition. So two factors are contributing. One is decreasing of surface. We know what our accretion rate is in the pond and the marsh.
our measured DIC efflux rate. There are, of course, issues with the next thing we wanted to do was actually a meter deep to the sediments and ask two questions. What are the sources, reactivity, and turnover times of slow cycling carbon? SOC. How did disturbance affect carbon? And this work was primarily done by Sharon Luck for her dissertation. I'm just going to show a few highlights. So one of the ways we characterize matter so thermal properties. Oh my goodness. Stay here? Okay. Stay, all right. I'm not going to touch anything. I'm just going to. Sorry. So we used thing. So in the marsh, the dark symbol, the vascular algal material is indicated by our hydrocarbons, and here we're using a ratio of even long chain hydrocarbons. Vascular to algal ratio in the pond surface sediments compared to the marsh. And so this suggests that the marsh is accumulating plant-derived organic matter in the surface horizon. And this has gone to 0, but I'm, all right, real quick. All right, so the next thing we can do with this thermal technique is then to look at the isotopic composition of carbon that burns off at different temperatures, both in terms of its 13C and its 14C. And so I'm not going to go through this whole figure here, but what you can see in the surface of the marsh on that side, all of those numbers are 13C values, and they're consistent with marsh organic matter. We see a very similar thing in the surface of the minus 13. Pretty confident that most of the organic matter in these soils is marsh derived. Now, the overlaid, these gray shadows, we have our thermal proxies. So this is more reactive organic matter and less reactive organic matter. We look at the 14C, and we see that in both the ponds and marsh, the less stable, the more reactive fractions, the lower temperatures, have modern fraction 14C values, and the less reactive fractions have less than modern. So this means that the pond surface horizons are accumulating low carbon with low reactivity that's derived from marsh grasses. So this is suggesting that we have reworking of soil organic carbon rather than deposition. The take-homes here are that the changing environmental conditions associated with ponding result in decomposition of this very. And what's relevant to this session is that this remineralization is then contributing to vertical and lateral fluxes. The last thing I'm just going to show you real quick is that we tried to calculate turnover rates of this slow cycling organic carbon pool. And we only could do this for the marsh because we have some violations of our model assumptions for the ponds. But essentially, we have a two-pool model that best describes our data, with the first pool turning over around 640 years and the second pool turning over around double that. What this means is that some fraction of old carbon is constantly going to be coming out of the marsh and contributing to that this old carbon is not inert, but it does turn over. And so where I want to leave off, and thank you all for your patience, is that soil organic carbon is really vulnerable to decomposition. All stages recently produced losses, but we still don't fully understand the carbon produced by decomposition via the relative contribution. And lastly, what I think is a big gap in our understanding, that in order to assess change the composition of really big challenge. My collaborators and lots of
Thank you, Amanda. Sorry about some technical difficulty. Should we just move on, or? So maybe have a quick question for Amanda?
Yeah, hi, Scott Gifford from UNC Chapel Hill. That was a great talk, and my question is, sorry if I'm too close to the microphone. You know, it used to be we really kind of pictured fungi in the food web there, and so can you track if there's a intermediate step in between that in terms of when it falls from leaf, or is that process not really dominant or hard to see?
So in terms of like our invertebrates shredding it and eating it, and that's making it more decomposable?
Yes, yeah.
So we didn't do that in these experiments. So I specifically excluded that, but that would actually speed up the rates of decomposition. So yeah, that would be an important factor, and that would be an more important factor in, say, the tidal creeks in any of these ponds we worked in, because they go anoxic. Really good point.
We're still working on the technical difficulty. So another question, maybe?
Incredible data there, Amanda. So how much of the old carbon do you think is breaking down? I mean, obviously there's a limit, otherwise you wouldn't accumulate. Yeah, what do you think?
Yeah, and I think it's going to vary by system, and I think it's going to vary by what kind of carbon is in the system. So what are these mineral organic matter interactions? How we change the environmental conditions, what does that mean for the microbes there in order to make it? I think that's really understanding those controls. That didn't answer it. Oh. Yeah, and I think with like sea level rise or disturbance going on in the soil column, that could.
Question?
Thank you. This was really cool work. I was interested in the decomposition, if there's some understanding about the role of fungi, especially because these are really like complex organic material, and I'm wondering how much is known in the salt marshes.
I think that is a really exciting field. I don't think we know very much. There's been a few really cool papers that have come out so far suggesting that fungi.
Okay, so let's move on to the lightning talk. So we have Amanda Mahadevan from WHOI, senior scientist at the PO department. So he's gonna show some 2 slides. Thank you.
Okay, so I'm going to talk about how flow or hydrodynamics in marshes and seagrass beds can induce instabilities, which then feed back on the morphology of these systems. So this is a photograph from, it's a Google Earth image of a marsh. Let me start by acknowledging my authors too. All right, so the question we're asking here, we've heard a lot about ponding, and the ways that marshes are responding to sea level rise is differing. So on the top left, you see an image of Barataria Bay, and that region in Louisiana, there's a lot of ponding. We've just heard about ponding. And the marsh is being lost because of sea level rise and subsidence and many other effects. And in the lower right image, you see another marsh where you see that channelization is very effectively draining the marsh. So the reason we want to try to compare these 2 different systems is we think that channelization is good. It helps to drain the marsh and makes the marsh more resilient to sea level rise, versus ponding results in the failure of the marsh, as we've just heard. Okay, so the question is, how do we generate channelization, or what are the natural ways in which we can do that? So this is a study site that we are looking at, and this region, we saw these channels develop. So the water in this creek is going up and down, and as the water goes up and down, it's flooding the marsh, and these channels are eroding headward. So that really confounds us, because we say that erosion occurs when the fluid stress exceeds some threshold, and then that leads to erosion. But at the very tip, or the landward tip of the channels, the flow actually comes to rest. So there is no flow. So how is it that these creeks are eroding headward? And using these historical images, you can calculate, we calculated, georeferenced them, and found that these creeks are extending headward at the rate of about 2 meters per year, which is really pretty fast erosion. So we want to know how this marsh is getting channelized. So we used, our ideas came from a very simple model that we developed. It was a model where the flow and the erosion interact. And we said that we coupled it with the 3 kinds of medium: the flow, the marsh itself, and the erodible material. And we said that the hydraulic conductivity of these channels depends on the porosity. So when you start with a medium which is fairly uniform and you pass flow through it, you generate a little disturbance, and that disturbance keeps growing because the porosity in that region is increased, and that drops the increases the hydraulic conductivity, drops the threshold for erosion, and that leads to channelization. So this is one way in which we get channelization. We make that region where the channel is more susceptible to erosion by forming the channel. And that kind of runs away that effect, and you start with a uniform distribution, and the flow all ends up being mostly in the channels and hardly any flow outside the channels. So the question is, how is this happening? How is the erosion threshold dropped in the natural marsh? And some studies which my co-authors have been doing, they've been looking at these marsh regions, and especially the tips of the channels, and they find that they're full of crabs, and the crabs are creating this porosity and burrowing, a disturbance which drops the erosion threshold and allows these channels to progress headward. I'll tell you about another study very quickly in a minute, is looking at submerged seagrasses. And this phenomenon that we try to explain is monami. So when you have a unidirectional flow over a seagrass bed, the entire grass bed starts waving. And that's because of a flow-induced instability. You get vortices at the top of this grass bed, which causes waving. So we developed a model for that, in which the drag of the grass is creating a reduced flow in the drag canopy, and most of the flow is above the drag canopy. And that shape of the grass is due to the drag forces, and that drag is put back into the flow. So here is our simple model for this. And you see that we generate this vortexes at the top of vortices at the top of this canopy. And these vortices bring about exchange with this canopy. So we want to know how you can enhance or reduce the exchange, what controls the exchange of this canopy. And so we can add tracers now to this, in this case particles, and you can see that these flow-induced instabilities are pulling material out of this canopy. So we want to take it to the next stage and put more realism in this model and couple it with waves and try to look at what those rates of exchange are across the canopy. Thank you.
Next speaker.
Oops, thanks.
We have a next speaker, Tzortziou from LSU, Louisiana State University. She's going to talk about tropical cyclones, cumulative cyclones, cumulative control on regional carbon fluxes in Everglades mangrove wetlands.
Good afternoon, everyone. My name is Tzortziou from Louisiana State University. I think I'm going to talk about a little different carbon POC that's represented by the mangrove litter fall. In some way, probably you can see, you can feel, and also you can hear. At global scale, the tropical cyclones are highly pulsing natural disturbances and have major effects on the mangrove structure and function. This map shows the overlap of the global mangrove distribution with the tropical cyclones. One of the hotspots is over there, the Gulf of Mexico. Okay. So due to these different disturbances, current mangrove carbon budget estimates still have large uncertainties. And to estimate this budget, we need to assess the production of mangrove that include the litter fall, wood, and root. All this production could be buried in the soil, could be released back to the atmosphere, also could be exported through the tidal cycles. Most of these components have been accounted in global carbon budgets. And however, there's still about 50% hasn't been accounted in this budget estimate. So in this work, we use the litter fall as a metric to constrain this carbon budget. And a little background, the litter fall deposited on the mangrove floor can be potentially stored as soil litter and also could be exported by tidal cycle. When the hurricane occurs, there will be the high-speed wind will deposit more litter fall. We consider that part as the excessive litter. And meanwhile, the storm surge triggered by the cyclones can also export more litter from the forest to the estuary. So, however, we don't know the magnitude of this excessive and exported during this hurricane event. So, the first question here, we want to quantify the magnitude of these two parts of the litter fall. The mangrove forests in Florida Everglades are exposed to high tropical cyclone frequency as the map shown, and the 3 long-term study sites established along the Shark River and the monthly litter fall collected in these sites. So we use this long-term data to estimate this excess litter. We identified there are 8 cyclones did deposit excess litter. So overall, we find that this significant excess litter production varied among different study sites and different cyclones with different strengths. And one single cyclone can deposit about up to 80% of total annual litter production. And for the exported part, they can export more than 50% of this litter to the estuary, especially during the strong hurricanes such as the Wilma and Irma. And based on this estimate exported, we also include the export by the daily tides. So we put them together, we estimate this POC export in this region is comparable to the carbon barrel rate and dissolved in organic carbon. And our estimate constrain the carbon budget in the aggregate region, and that compared to this global estimate, we constrain that. So the second objective in this work, we want to say, can we modeling or estimate this using a kind of simple model? So we use this energy, which is to assess the energy of the cyclones, which consider the wind speed, cyclone size, and the trajectory. And so we know that common sense, the excess liter could be related to this energy, also the distance from the cyclone to the study site, and also how long the mangrove forest will be exposed to this disturbance. So we put these three components together and define this pulsing index. And then we put this index and excess liter together. We find that this excess liter highly correlated with this pulsing index. So we propose to use this statistics model to determine the mangrove role, to determine the hurricane role on the mangrove as a carbon sink or carbon source. So I would like to thank all these funding agency, and also thanks collaborator from RCU and FIU. So please check our paper for more details. We look forward to hear from you with any interest and concern. Thank you. Yeah.
Thank you. Next, we'll have Xinping Hu from Texas A&M Corpus Christi talk about the tropical estuarine budget under different hydrological conditions.
Texas has been so dry, even my slides cracked up. Anyways, I don't know whether you guys noticed this morning and this afternoon, all these plenary speakers, they were all holding on to this terrestrial organic carbon. So here I would like to introduce this subtropical estuarine carbon budget. This is actually the first carbon budget built up for the northwestern Gulf of Mexico. That's Jaime was my second PhD graduate, and he finished his defense. And before walking, he actually abandoned me for the commencement, went back to China to his girlfriend, and then who became his wife later on. And then thank my colleagues, longtime colleagues, Paul Montoni and Mike West, and also Corey Sterick, ODO graduate, my technician. So just to show you the Texas coast. Okay, so this is part of the northwestern Gulf of Mexico. When people talk about climate change, people look at the time, right? But if you talk about climate change, what you can do is to stand at the Louisiana-Texas border and look down the Texas coast. Because if you look, this is the precipitation gradient, roughly matches the latitude from the northern Texas coastline and then the Texas-Mexico border. So decrease of fresh water, precipitation by a factor of 2. So red bar represents evaporation. So now if you look, subtract evaporation from the precipitation, so you can see that in the northern part of the state is mostly positive estuary. There's excess fresh water, but then if you move to the south, there is a gradually increase of the dryness. So actually from where I live is considered as semi-arid. And then if you consider another factor, consider all this fresh water coming into the estuary and the evaporation and diversion. So that's what we call this freshwater inflow balance. And that is even more extreme because precipitation decreased by a factor of 2, but the freshwater inflow balance is a decrease of 100 times. So very extreme. As a result, estuarine water in the northern part of the state, residence time is roughly a couple weeks, but then if you go to the south, it can go for more than a year. So salinity in the estuaries can also be variable, but then in the northern part of the coastline, in low teens or lower than 10, but then if you go to the south, Laguna Madre, which is the yellow shaded area, so that area salinity is usually about 40. Some part of the tributary can go above 70. So people have to dilute the water to measure the salinity. So Here, just a brief introduction of the work. Actually, was published in the journal Water Research earlier this year, a couple months ago. And we basically surveyed the mid-Texas coast estuaries along this hydrological gradient. 4 estuaries, so from the north to south, Lavaca, Colorado estuary, San Antonio estuary, Mission Aransas estuary, and Nueces estuary. And we basically looked at water chemistry, river input, and then also we combined with the agency collected data, USGS, Texas Commission on Environmental Quality, and also Texas Water Development Board. We looked at the carbon budget in these 4 different hydrologic conditions: drought, flooding, flood relaxation, and hurricane. So just to show you that this is Hurricane Harvey track, landed in Texas in 2017 in August. On the right, this is a very basic figure, so I had to use the log scale because otherwise the lower coast rivers will look like a mass spectrum. So that's the northern part of the coastline. The Guadalupe goes to San Antonio estuary to the left, and then Aransas Mission estuary go to the Mission Aransas river, go to the estuary, and Nueces River goes to the Corpus Christi Bay, basically. So just to show you, that's what we built. The land-ocean interactions model in the coastal zone. We quantified the CO2 fluxes. We quantified the water mass exchange between the estuary and the coastal ocean based on water budget. And we looked at carbon deposition in the estuary and also the input of organic, inorganic carbon into estuary from the rivers. So then the mass balance indicates the leftover is from the lateral exchange, DIC, and the TOC. And just to briefly show you that these are the 4 different conditions: drought, flood, relaxation, flooding, and a hurricane. You can see there's a lot of variations in these terms. Just to highlight a few items, CO2 exchange increased substantially between drought and the flooding period and the hurricane period. And also, freshwater-rich estuary has a lot more negative net ecosystem metabolism. And then also the lateral exchange of the carbon for DIC and TOC from the tidal wetlands is much bigger than from the river. And just, finally, this is the carbon budget. So the only thing I want to emphasize is you can see the TOC and DIC from the tidal wetlands is a lot greater than that coming from the rivers. And then finally, acknowledge my funding agency colleagues and also the data from the local agencies, federal agencies. Thank you.
Thank you, Xinping. Next, Yinchuan Mu talk about the potential for CO2 removal. We're enhance the weathering in the Amazon River to ocean continuum.
My name is Linquan Mu. I'm a postdoctoral fellow in the University of Rhode Island. And today, I'll talk about a hypothetical CO2 removal experiment by adding alkalinity in the Amazon River watershed. First, a little bit of a background of our study site. So the Amazon River is the largest river in the world by discharge, and its outflow brings massive amounts of fresh water into the tropical North Atlantic Ocean. And as the river water mixes with the salty seawater, and it will create this thin layer of fresher water that's sitting on top of the ocean surface, and we call it the Amazon River plume. And this plume water will have a lot of influences for the ocean surface. And one of the many things that this plume water can do is to dilute the seawater and control the CO2 mechanism in it. For example, if we look at the figure here, if we take a river end member and an ocean end member and we mix them, and we use the TA and DIC as conservative tracers, assuming no biology, then we'll get a mixing curve that's a straight line. And we can then calculate the PCO2 from TA and DIC, and we get a PCO2 mixing curve that's starting from a river CO2 that is really high, and it decreases sharply with the salinity, and it reaches this minimum below the atmospheric level before it climbs back up again towards the ocean end member. So essentially, what we're trying to do here is to demonstrate that this kind of a PCO2 mixing curve is necessary for us to understand the effect of the river outflow to the offshore PCO2 as well as sea air CO2 flux. And in order to get a mixing line like this, we need direct measurements of TA and DIC at the river mouth, which we do have from a previous cruise. And we will use this as the baseline scenario, and we hypothetically add alkalinity at the river mouth, and we'll monitor how the offshore CO2 will be changed as a result. So here, the plot on the left, we have a PCO2 mixing curve calculated for a variety of perturbation scenarios in colors, in addition to the black line, which represents the PCO2 mixing curve baseline that we already seen. So we assume that the added alkalinity will react fully with CO2, and there's no secondary precipitation of calcium carbonate. And we also assume that the biological effect on CO2 will be remaining the same from the baseline to perturbation scenarios. And so the take home from this plot is that the PCO2 will be decreased as we add alkalinity in it. And the more alkalinity we add, the more CO2 it will be reduced by. And also note at the higher end, which is close to the ocean end member, the PCO2 barely changes. That's naturally because the river influences doesn't reach that far. So now we focus on a specific perturbation scenario, which is +10 in red. And we calculate the difference between the red line and the baseline, which is the black line. And we apply this difference on the PCO2 that we originally used to calculate the baseline flux numbers. And we get the flux map in the middle. So this is how much the flux has been changed as a result of alkalinity addition. So we can see for different parts of the plume, the reduction of air-sea CO2 exchange is different. And we can also pair it up with this original flux map. And we can see for most of the plume area, the CO2 flux change has been below 0.05. And there are areas which have a higher influenced by the alkalinity addition. Now, we take a step back to see how we can detect the alkalinity we add in the watershed. And to do this, we have the TA baseline in black, and it has a mean and a standard deviation, which represents the natural variability. And we add alkalinity to get this red perturbation curve. And then we perform Monte Carlo simulations and the students t-test between the perturbation scenario and the baseline scenario. And so there's more details in the poster, but the take home here is that the p-value that is below 0.05 is the condition where the perturbed TA can be detected with a larger than 95% of confidence at the river mouth. So just to quick sum up in this Q&A format, will the TA addition in the watershed reduce PCO2 in the plume? And our answer is yes, but it's salinity dependent, and the PCO2 reduction will be more pronounced at low to mid salinity area. And similarly, will the TA addition in the watershed change the CO2 flux? And it's a yes as well. And similar to the previous question, it's depending on salinity. And an MRV question, so can we, how many alkalinity do we need to add in order to get a detectable TA change relative to the background variability? And in our case, it's about 10 units alkalinity addition. and if we take samples, like 30 samples, but of course, if the background variability changes, the number will change as well. So I know the poster session has ended, but if you are interested and you haven't had a chance to look at it, feel free to scan this QR code or reach out to me. Thank you very much.
Thank you. We have the last lightning talk by Jake Supino from Boston College. And his topic, constraining salt marsh carbon and nutrient cycling using autonomous biogeochemical measurements at the 7 Mile Island Innovation Lab.
Hi, my name is Jake Supino. I'm a PhD student at Boston College working with Hillary Pilevsky and Kristen Fogren. And I'm going to be telling you in introducing our work doing salt marsh carbon cycling. So not to beat a dead horse, but to beat a dead horse. Salt marshes are really productive systems, and they provide a wide variety of ecosystem services. The one that I kind of want to emphasize right here is this one, because I think it does a really good job kind of humanizing it in layman's terms for people to understand. But this also really matters, especially in places that are really susceptible to sea level rise. And New Jersey's like definitely up there. So there was a recent report that just came out, and it said that sea level will rise by about 1 foot by 2050, which you can see here will completely inundate the marshes in southern New Jersey, and basically make it incapable of providing those ecosystem services. So my research site is the 7 Mile Island Innovation Laboratory, also called SMIL. It was established in 2019 as a collaboration between the U.S. Army Corps of Engineers, the Wetlands Institute, and the state of New Jersey as a test bed to advance dredging and marsh restoration techniques. This was chosen because it is really close to a coastal community that relies on these ecosystem services. It's the home of the Wetlands Institute, which is like right there in the middle. And it is bisected by the New Jersey Intercoastal Waterway, which is required to be dredged by the U.S. Army Corps for federal and state travel. So historically, there has been a bunch of beneficial use projects. So we can kind of like see like legacy salt marsh sediment placements. But about 50 years ago, this was not possible. And instead, the removed sediment was basically taken upland or released into the open ocean. But through this new idea of using sediment as a resource, it's called beneficial use sediment placement. They've been putting the dredged sediment on top of the marsh so we can keep pace with sea level rise. And some of the new studies in this area have really only looked at sediment transport and wildlife, but we haven't looked at the biogeochemistry yet. That's kind of where we come in. So our goal here is to quantify baseline rates of carbon, oxygen, and nutrient cycling, and to monitor responses to episodic events and understand long-term change. So we have that kind of like natural perturbations, but we also have the anthropogenic ones as well. To do this, we have 3 water quality monitoring sites, and this is what like the stations look like. And we have 2 multi-parameter water quality sondes at each site and a few other instruments. And this is some of the parameters that we're taking here. So as an example, what we have as in terms of our natural observations, we can see some seasonal changes. This is fall going into December. We have weather events, which you can see in the gray box. That's a nor'easter that came through. And as we zoom in, we can see those diurnal cycles. So on the more anthropogenic side, there is an upcoming dredge happening in October. And we are placing 5 floats around Sturgeon Island, which are those yellow circles. And what we're going to do is we're going to evaluate the ecosystem baselines. We're going to have them out during the dredge placement so we can monitor what's happening during that. And then afterwards, as the ecosystem returns to homeostasis, to really understand the metabolism and the productivity. So thank you for listening. I'm really excited about this project. It's important to find out for a lot of other beneficial use projects in the area and around the world. So if you are interested, you can get in contact with me here. Thank you very much.
Thank you. To conclude our part of the so next we're going to have the panel discussion. So I'll invite all the panel speakers up to front. All right, so questions for people. I guess I can start with some kind of common thing. I have a slide, but I think I can just talk through it. So there's a couple of points that the plenary speakers mentioned. So, you know, the one major question is what are the major unknowns or major steps we should do next to better constrain the lateral fluxes. And this is also the question that Galen mentioned, you know, what may be the next step that, you know, how we do this so that we can scale up the estimate, you know, not simply taking average. I can just pass my mic to the plenary, their own take on what may be the next step.
Yeah, well, that's loud, sorry. Yeah, I think just from our conversations, I think isotopic measurements could be helpful for understanding, like what Aleck was saying earlier, not just sources of where the DIC is coming from, but also like the timing of if it's recently fixed or older sources of carbon. And I expect that in our previous work in peatlands and recently restored systems, sometimes the older carbon gets more exposed, and we talked about that with the burrowing too. So I think that could play an important role. And then I also really liked Isaac's work with like moving out. I think you'll probably talk about that more, but not just staying in our tidal creeks, but moving out and watching it and going out to, you know, farther out of the marsh and seeing how it actually disperses is a really important area to go into next. So yeah, I don't know, Kevin, you want to go? Oh, yeah, you have them.
Yeah, so to add to that, I think it's going to be very important to examine high frequency and extended time lateral fluxes in the context of the covariance data to provide us with activity, respiration, processes, the source sources for the carbon. So as Patty is doing, and some other started. That's going to be very important. I think another step that we can take is to look at some of the longer term data sets and understand how we can interpret shorter term data sets in order to Isaac, for instance, alone has about 100 of them, and so I think there is a lot of high quality data out there now as compared to in the past, and
There's a question from Bob Anderson on the line, but on the Zoom, but I don't know. I guess you were kind of cycling through the plenary speakers on the fate of understanding export. Should we wrap that up or get Bob's take? Oh, I didn't say it yet. I was wondering if did you want to have your chance to talk about the Aleck's original question before we move on? Sorry to interrupt.
Yeah, I can make a quick comment on that. I think, like the Blue Ocean community, you make these measurements of sinking rates, and they're always looking for these easier to measure indicator, let's say chlorophyll, a global map of chlorophyll that you can get from satellites. And they're not trying to use chlorophyll to kind of hold your data set together and build extrapolation. So, I mean, that's the ideal situation, I think, that you have something we can get from satellites, like, let's say, tree coverage, or tree heights, or, and now we have these global maps of, let's say, soil carbon density or soil accumulation. Those are the things that are well established, so we kind of need to build relationships between the hard to measure things to the ones that are kind of easier, and presumably make those upscale exercises. But I think we're really far from that, because there is many problems that we need to solve before we build those relationships. They're not showing up on the data sets we have, because I think we have issues of scale, both spatial, temporal definitions of how we express those things. And also, I would say the techniques, the approaches you're using to measure those things. We don't even know if they're comparable, like Alex showed some beautiful data comparing techniques. And, they're in the same order of magnitudes, but that's not enough. You're gonna upscale things in a real nice way. So right now it's average, that's what we got.
Okay, here's a question from Bob Anderson. And, unless Amanda, did you want to respond?
No, I don't have much to add.
Oh, okay. So, and there's just a request to hold the mic a little closer when you're responding. That goes across the board. So the question is, sulfate reduction was mentioned this morning as an alkalinity source, but where humans have disturbed marine wetlands for agriculture, aquaculture, et cetera, oxidation of sulfides produces sulfuric acid, and this acidification has been a problem locally. So the question is it significant in terms of the amount of alkalinity produced and exported by wetlands globally? The oxidation of sulfides producing sulfuric acid, is that a global problem?
Yeah, that's a great question. So the person with the question, Bob, is talking about our acid sulfate soils. Those are widely distributed globally. We think there are about 250,000 hectares of drained tidal wetlands in the United States, maybe about 13% of the total. And they do release quite acidic water. And so, yeah, we have a famous example at one of our study sites at the Herring River at the Cape Cod National Seashore. And yeah, so I would suggest that they probably are a significant part of the budget.
Yeah, let me also add to this discussion, because Australia has been I think the country has been greatly influenced by this acid soil, sulfate soil. But then another thing we found in Texas estuary, that is when the river inflow was really low, and we saw this really enrichment of sulfates that cannot be explained by the mixing between river and the ocean. And similar phenomena was observed in North Carolina estuary. That was back in 1980s, but I don't think anybody caught up, any work after that. So definitely sulfur is one issue, haven't put a whole lot of attention to this.
So you have tropical Savannah to a little warmer Sapporo Island, and then come up to Cape Cod, is a little colder. And here we have a western boundary current system contrasting to eastern boundary in the California current system. So what do you see the difference or lateral transport of these different contrasting ocean conditions and climate gradient? I would like to hear your view. to all of you.
All right, I can start a little bit since I study shelf ocean exchange for a while. So, so this is the last, the motivation slide I showed. So I think this the core question, which is the fate of all the carbon export from wetland, right? So as I mentioned, the alkalinity export as the kind of the blue carbon credit, how we calculate that's the net. But there's this assumption, if I'm not mistaken, which is assuming it is equilibrated. The DIC will equilibrate with air, and so by the time is exported, make to the open ocean, it's pretty much the CO2 or DIC is removed from the system. Only the alkalinity left import. But we all know that's not the real case. We actually don't know how much the DIC being degassed in the coastal or shelf water. And then this to the question, we don't know, but I'm assuming the upwelling system is going to be very different in terms of export of carbon, shelf carbon to the deeper ocean versus, yeah, right. And then west boundary is much wider, and then the export could be more because there's a longer, there's a uptake of CO2 on the shelf, and then that's being processed that can be exported to the deeper ocean.
Yeah, listen, I really wish I had an answer, but, I think the path for that, Patty was laying that path, with just multiple locations where you can put edge covariance. So that type of network is essential. Like the Blue Ocean community has been fighting for that for decades, and you guys have been quite successful. It's never enough, but you got quite a few buoys around the place. You got gliders traveling, everywhere under the ice. So I think what we need for this community is actually develop those networks. I realize there is many people from agencies here. Now, I cannot get your money, but, I'm in Europe. But I think that's exactly what you need, have that type of station that, look like those oceanographic buoys, like, HOTS, like, Aloha, those stations, but doing the blue carbon ecosystems. And that's really hard and expensive. There is a few of those individual initiatives like Kevin's and Aleck, but, that's the direction, and you got to have those in strategic locations, not only in the U.S., you got to go beyond, right? We're talking about, some tropical hotspots like Kenya or Indonesia, or the Amazon. So those places are really hotspots. So I would love to see that happening with the blue carbon community, down the road.
Is it okay if I ask a question? I'm holding the mic, so why not? So I really appreciated all the talks. And I guess this question is inspired by something Kevin put up, but might be something a number of you want to answer, which is that this project Vera that verifies carbon uptake has verified a gigaton of carbon. Is that in the blue carbon only or across carbon? And then the follow-on that's more general is, like, how mature is this? You can't count marsh carbon that's already was happening anyway. It's really the additionality of a human impact on a marsh. How ready are we to quantify that and provide that as an estimate for people who want to buy it or so?
Yeah, I can answer the first question. That was the total for that organization, so that it does include lots of different other kinds of projects besides blue carbon. I don't know what the total is for blue carbon at that particular organization, but yeah, it's a good question. I know that particular organization and methodology is quite detailed. Ultimately, it's very conservative in terms of how much crediting a project can get, probably an underestimate by a significant amount. And that's on purpose to avoid the criticisms of overcrediting carbon projects. I think we do need organizations. I don't know what organizations we need governments perhaps to step in and evaluate and modify as necessary, and ultimately put their stamp of approval on carbon crediting for broadly, not just blue carbon, because if we don't get that right, then the perception is poor regarding carbon credits, and all of that investment towards ecosystems will go elsewhere. So I think that particular method is quite rigorous, but I certainly wouldn't claim to know whether projects or any of those projects are of the value that the credits that they were given.
Yeah, I can add to that a little bit. I helped actually co-author the greenhouse gas offset methodology for California wetland restoration, which has been adopted by this voluntary carbon market, and tidal wetlands were included in that offset methodology, but it hasn't been applied yet, as far as I know, to tidal wetlands, only to freshwater peatlands and those ecosystems. But it's also very conservative, like you're saying. Additionality is a really huge issue, and lateral fluxes are, of course, not included in that. It's difficult with, again, I think I mentioned this earlier, with tidal wetlands too, because permanence is a really big issue. You need to be able to have it guaranteed staying there for at least 100 years. And so with wetlands, with sea level rise, it's hard to say that in some cases. There are a lot of problems with it.
Add briefly to that, alkalinity in the ocean, as has been said several times here today, will easily meet the permanence requirement, 1,000 or thousands of years. And reductions in methane emissions as well have an inherent permanence. Those can't be reversed. So there are aspects that easily do meet this.
Great. Also to follow up a little bit on that too, from, sorry, what Isaac was saying too earlier, and I think what we've learned from the EdiFlux community is the sooner we are coordinated and collaborative, the better it is for the whole community. So I think one of the really key things with that community was having platforms for data sharing and data standardization. So if we are, it does seem like it's an emerging field. There's more and more people collecting these data. It makes sense to have that set up somehow and have, funding sources that are dedicated to that standardization. And the other really great thing with that is that you can, like Kevin was saying, if you can take these shorter term data sets and use the longer term ones to help upscale those ones, and yeah, just more inclusive.
Sorry, this wasn't on. So I'll just repeat the basic question. I think everyone in the panel can hear, but for people online, what's the role of satellites in getting to the quantification of some of this?
Hi, it's Suzanne Craig, NASA GSFC. I just wanted to kind of add on and amplify to what Maria Giorgio just said about remote sensing. There's a lot of opportunity and perhaps unexploited opportunity to use spaceborne sensors. But there's also, I think, a very untapped opportunity to use unmanned aerial vehicles to make all sorts of spatial surveys. that includes not only the reflectance of aquatic and terrestrial systems and wetland systems, but also of the evolved gases that are climate relevant. All of that technology is now commercial off the shelf. It's highly affordable. It just needs the will and the coordination of the community to fully exploit it. But I just wanted to make that synergistic comment with what Maria just said.
Yeah, it's a good question. I can answer a little bit. We do have a project using dense time series Landsat images in order to detect phenology over years and breaks in the phenological cycle so that we can detect changes in condition of wetlands. This is led by Tzortziou at University of Connecticut, and can also detect soil moisture and water on the platform. And I think it looks like we're going to be able to predict fluxes with some accuracy based on productivity and hydrological exchange. And I think those are both detectable from space for sure. Need a lot of on-the-ground measurements to test those predictions, though.
So just add to that, I think we now can maybe start to identify some of the drivers, and then we need to, if we identify some of the drivers, I envision we can use proxy to model those drivers, including, remote sensing parameters. That will be, my hope is that could get really the larger scale up, and do a more robust estimate.
Maybe this is, so I don't do satellite at all, but one of my understandings from working with some folks is that one of the big challenges is filtering out tides and having enough data resolution that you can do fluxes, that you can identify these signals despite and account for what's being exported via tides or what you're not seeing due to the tides. How do you consider tidal filtering?
So I don't do remote sensing either. I provide wetlands and carbon information to people who do the remote sensing. But so filtering for which kind of features? Do you mean
when it's inundated, it's not inundated? How do you have the satellite? Is there enough satellite resolution to know what you're measuring when you're measuring it, and filter out the times when the platform is flooded and you're not getting, say, full GPP, right? And you're not, your fluxes are changing, right? Because you're totally inundated or you're not inundated. And so my very, very limited understanding is that you need this high satellite product in order to, Maria, you're nodding, so you know a lot more about this than I do.
Yeah, so tidal information on tidal stage can be integrated in some of these tides, and you can say when a satellite image was collected at exactly what point of the tidal cycle. And this has been used in previous studies to actually compare high tide, low tide scenes and see how much tidal export impacts DOC dynamics, for example, in subestuarine waters. So even with, this was done with high spatial resolution sensors, and the problem with these sensors currently is that we have them every 2 or 3 days, even when you're looking at constellation of 4 sensors right now with the 2 Landsats and the 2 MSIs. But in the future, NASA is working on a concept for a geostationary mission, GLIMMER, and Suzanne is going to be discussing about GLIMMER tomorrow. And this is one of the reasons why we wanted to have a relatively high spatial resolution on a geostationary platform to be able to look at these impacts of tides and other physical processes on carbon export and biogeochemical processes in general in coastal waters.
I'd love to add to that too. We just had a paper come out of Eden Landing called Miller et al. In remote sensing who addressed this exact question using Landsat, actually. And so Gwen's a student of Irina Dronova at Cal, and she developed a model that could use like previous data and also things like drought indices, and created this basically this envelope model that could help you pull out the phenology of the system, even including dates that were inundated.
I have a question for Isaac from this morning. We didn't get to it on the beforehand. You mentioned that the impact of salt marshes is predicted to increase under climate change. On the U.S. East Coast, there isn't much room for marshes to retreat under sea level rise, given the human-built environment. Is this an issue on a global scale? This is from Tatiana Reinerson at URI.
Yeah, there you go. Sea level rise. I can see how that you can have different perceptions. I think yes. I don't know enough about the U.S. East Coast, but I understand there is not much space behind the marshes, right? There is nowhere for them to go. But there is many other places, with lots of marshes, including the Chinese coastline, where the salt marshes are growing like crazy, right? They are growing very quickly. They're replacing mangroves. So, I think they're probably thinking about, I showed a plot that makes a prediction that carbon accumulation in sediments is going up with climate change. Right. So that's the graph I showed, and that prediction is that on a global average, the models available, they actually show that on average, those places are going to grow rather than shrink if you can isolate climate change and if you consider that accommodation is space, right? So many places are going to suffer, there is nowhere to go, but other places are going to expand. That's going to be variable everywhere. But my understanding, and correct me if I'm wrong, that's not really my cup of tea, but that's what I get from the colleagues making those global models. On average, you expand the area of those systems. They grow.
I ask a follow-up question for you. So if you have a switch, so you're predicting a switch from mangroves to salt marsh, and so it's in carbon burial due to the switch in the assuming, am I going? Oh, sorry. Here we go again. Assuming a warmer world, you're going to have higher rates of production and higher rates of respiration, right?
I think that's a lovely question. I don't think we really have a clear vision of that. I have tried to take sediment cores at the transition zones so a place like the U.S., I'm not sure about the U.S., but definitely Australia, the mangroves are killing the salt marshes, right? They are encroaching onto the salt marshes. You go to China, that's the opposite. The salt marshes are, getting the mangroves out of the way. So we have taken sediment cores at those hotspots where you have this transition, trying to look, okay, what's happening with that ecological evolution of those systems? And the data that I got, my experiences is that you cannot pick up, the functional, I suppose, or the rates. So it's hard to say which one is going to sequester more carbon in sediments. So the sediments that are kind of easier to handle at this stage, it's difficult. And if you want to go into the outwelling, the greenhouse gases, actually, I have no clue what is going to happen with those replacements, those ecological transitions happening everywhere.
Okay, I want to thank all of our speakers again for this fantastic and really comprehensive view of the importance of these different types of transport and all of the different ways of measuring it that we've shown today. So thank you all so much.